Good morning, everybody. My name is Luca Ottaviano, and I'm chairing this Wednesday morning session of the virtual conference of New Times. And I think it's a good time to start. So welcome to the conveners. And first of all, I'd like to thank the chairs of this conference, Antonio Politano and Carlo Cantalini, Professor Carlo Cantalini, and indeed congratulate with them for the outstanding quality of this virtual event. And I wish that this experience will be repeated in the future, maybe in a mixed configuration, live and virtual as well. I think that you, Carlo, and Antonio did a great job in putting together this audience and community and establishing this new format. Okay, my name is Luca Ottaviano. I'm a professor at the physics department of the University of L'Aquila, Italy. And I'm chairing this. I'm particularly I had the privilege to chair this session in nanobiotechnology and advanced material for nanobiotechnology. Okay, so the thing is that living matter is the ultimate material. Living matter is the combination by evolution of nature of functionalities of materials and especially for what concerns the sensing, the actuating, and the functionalization of materials due to special functionalities. So the nanobiotechnology is also the ultimate field where the cross-disciplinarity is of pivotal importance. In nanobiotechnology, we must have the contribution of physicists, chemists, chemical engineers, mechanical engineers, biologists, of course, and also computational science. So it's a particularly exciting field, and I think it's very much suitable a session of nanobiotechnology in once someone is talking about new materials and new functional materials. The goal of nanobiotechnology and the materials in nanobiotechnology is interfacing with living matter, so sensing issues and actuating issues, and also issues relating to therapy of living matter. And also it is important the mimicking of living matter, and to understand the behavior of living matter, it is also very much important to understand the behavior of water at the nanoscale in confined structure. We will address all these issues and aspects of the various aspects of nanobiotechnology in this very interesting morning session. I'm really honored to chair this session because there are outstanding speakers. So I think the quality of this morning session is really outstanding, and we can almost start. I think we can start in 1 or 2 minutes. So I'm very pleased to introduce Francesca Santoro, and I was pleased to know that she's been included in the list of the 100 women who are changing the world. So I think she's changing the world because of her professional activity, and so we're very looking forward to listen to her contribution, which is essentially in the fields of biomaterials. Francesca graduated in biomaterials in 2010, and then she moved to outstanding institutions all over the world, in particular Aachen and then Stanford University, and then she is the MIT ambassador for innovation and in the world. If Francesca, tell me if I'm wrong.
Hi, thanks for introduction. No, all good.
So, okay, excellent. So the title of the presentation from Francesca is Designing in Vitro Biohybrid Synapses. And I think you have 2-3 minutes extra time because of the lack of questions from the previous speaker. Thank you. The floor is yours.
Okay, do you see it correctly? Do you see my presentation? Can you hear me?
Yes,
well?
yes, it's fine.
Okay, perfect. So hi, everyone, and thank you, Luca, for this very kind introduction. So my name is Francesca Santoro, and I'm a principal investigator at the Istituto Italiano di Tecnologia in the center, which is located in Naples. And in our center in general is very much interested in the development of new materials for healthcare. So today I'm gonna talk a little bit about one specific research project we do in my lab, which is the tissue electronics lab, where we in general trying to design materials and platforms which can be interfaced with different tissues within our body. In particular, in this case, we're gonna see how we put a great effort in the last years in designing what we call in vitro biohybrid synapses. So one main point here is really to trying to create platforms which are microchip based. So these are devices and materials in general which exploit electrical fields to interface with different systems, for instance, with ultimately with cells from the brain. And we really try to tackle into the real problem of this interfacing, both on the physical coupling level, but also trying to design materials which can be smart in the sense that they can really electrically adapt to the cells of this organ. Why electronic materials? Because neuronal cells do have an intrinsic electronic activity. So when they communicate within each other in a complex network, they do this through electrical pulses called action potential. So this potential starts from one cell, triggers the delivery of certain neurotransmitter at the synapse, which is really the contact point between two cells. This neurotransmitter is received by the second cell, and by the activity of a very complex protein machinery, it triggers another action potential into the second cell. So why is this relevant? Obviously, we want to develop this kind of microchip-based technology, not only to understand and to study the physiological activity of a complex neural network, but also we are very much interested in giving a contribution and supporting possibly with implantable devices, brains in general, which have been affected by diseases or pathologies like neurodegenerative diseases. So it could be really an idea to integrate a chip into a non-functional area of the brain. And this integration, in our case, is really try to be done by tricking, in a sense, the cells and trying to design these microchips as similar as possible to neuronal cells. But what is this similarity about? So when we say we're really trying to make materials and devices in general to look like neurons, to act like neurons, means that we really work on two main concepts. The first one is really trying to pattern, to design, and to synthesize materials which could really recapitulate the environment of neuronal cells in terms of curvature. So you see these neurons have these very nice branches, and their local curvature can change quite dramatically. They're pretty soft, so we're trying to reduce as much as possible the mismatch between, the young modulus and the stiffness in general between the cells and the chip. And also we're trying to recapitulate a very dynamic and three-dimensional environment. On the other hand, and this is what we're going to see today, we're also trying to develop platforms which would also resemble functionalities of the neurons. Typical microchip can, just record or can send stimulus like electrical pulses, like to trigger, to modulate the electrical activity of cells, and they don't really adapt. So that it should be someone else that is imposing the protocol of the simulation and of the recording. Here we're trying to develop a platform which can resemble, can adapt, can interact with the neuronal cells in a more, let's say, smart way, by resembling the synaptic plasticity of cells, by exhibiting learning capabilities and the typical connectivity pattern, which is already been present in the physiological neural networks. So we get inspired by, when we design these materials, again, by the key point of the connection with the neurons, which is the synapse. As we said, this is a contact point where two cells start to interact, and then so this spot is really responsible for the short and long-term communication. In fact, neuronal cells do have this kind of phenomenon. So a short-term potentiation is called, which is, as I said, triggered by neurotransmitters being released from one cell to the other. And this kind of process, so this kind of very strong communication happens in the range of tens of milliseconds, while the more the cells start to talk to each other, the more they would keep talking over time. And this is also what is so-called long-term potentiation, which is a phenomenon that acts on a scale of minutes or even longer. And so what we try to do here is really to develop a platform which can replace one terminal of the synapse. So usually, as I said, two cells come together. We have the presynaptic cell, the postsynaptic cell. So the presynaptic cell is releasing the neurotransmitter because of an action potential. The postsynaptic cell is receiving the neurotransmitters, and which cause also an opening and closing of receptors, which are specifically binding the neurotransmitters. And so what we did here, we replaced the postsynaptic side with a transistor. In our case, so a transistor is a three-terminal device. In our case, we used an organic semiconductor. So this is a polymer which is doped, and we'll see in a moment how that works. And so what we had then, we had a functional coupling between the presynaptic part, which in our case were actually still biological cells, with a three-terminal device to a microfluidic platform. And we had these cells which just selectively released dopamine as a neurotransmitter. So sort of a kind of fake neuron. Instead of releasing a bunch of different neurotransmitters, we would just, let's say, lower the degree of complexity of the system by having cells only secreting the dopamine neurotransmitter. So why using organic semiconductors, and specifically why using organic neuromorphic devices? So these transistors, as I mentioned, are very, special in the sense that they can really resemble the plasticity of cells on the both on the short and long term. We use organic semiconductors mainly because they operate at a very low energy level. Being organic means that they're very much favorable and they are a good fit for biologically related applications. So these materials are biocompatible. And also when we define this, behavior as a neuromorphic behavior, this means that we can tune the memory of this device. So this device is practically a memory, like a neuron in our brain is, where different input can trigger different, let's say, states, and these materials actually allow for this very easily. How those materials work? So the material that we choose among the family of organic semiconductors is a PDOT-PSS. So this is a thiophene, and when in contact with an aqueous electrolyte, which has cations inside, the cations, when a voltage is applied, are pushed from the electrolyte towards the PDOT-PSS channel of the transistor, generating let's say a current into the transistor itself. So as you saw here in the animation, so we don't apply, in the case of neuromorphic device, a constant bias to cause this the field effect, but we actually apply a pulse, kind of a train of pulses, which would really, let's say, ideally move those charges back and forth, but in the reality, some charges are retained, and that is what causes actually the memory effect in our device. In terms of, so we talked about a current that is generating the device. It reflects a change in the conductance of the material itself, as you see here, and as is highlighted in pink. When you have this pulse that is generated at the gate of this transistor, you have a decrease in the conductance level of the neuromorphic device. And this is actually, this you can move back and forth, and this means that it represents a sort of a volatile memory. So now we wanted to emulate what we were discussing before, both the short and the long-term modulation. So what you have seen before, just when you have a pure electrolyte, you can just briefly modulate the conductance level of the channel. So it's a volatile memory and represents a short-term modulation. But now, when we have a dopamine in solution and we apply the characteristic oxidation potential of dopamine as a gate bias, we can induce the oxidation of dopamine, and during this oxidation, 2 protons and 2 electrons are released. So 2 protons are giving it a further push to the cations. They are moved from the electrolyte towards the PDOT-PSS channel. And this is actually what happens. So before, if we look at the conductance in pink, we still have what we so-called the short-term modulation, exactly what we've seen just when we had an electrolyte solution. But now we have a further decrease of the conductance given by the contribution of the protons generated by the dopamine that has oxidized in the solution. And this kind of interaction is actually giving a long-term modulation of the channel. So the value of the conductance won't get back to its initial value, but some memory or charge memory is retained into the device. And this is causing what we call a synaptic conditioning. Obviously, we have proved this for a number of concentrations of dopamine. How we have seen this being actually consistent and increasing with by increasing the concentration of dopamine solution. And then we move to coupling cells which, as we said, secreted only dopamine with the device. In red, you see dead cells. In green, you see live cells, and we made sure that the contact between, the artificial neuromorphic device and cells was good. And actually, we had a very high viability of cells. And also by electron microscopy, we visualized the interface, the contact point between the cells and the surface of the material. This is a cross-section done with the scanning electron microscopy in focused ion beam. And we could even resemble, the typical cleft interspace, which are in the case of synapses of few microns. And then we went to the, let's say, to checking, other than the cell viability, obviously, but that cells would still physiologically release the neurotransmitter, as you can see from this fluorescence labeling in the micrograph here, where we stayed in green for dopamine, and this meant that dopamine was there, and everything, let's say, was, let's say, the interface was still functional. And then we have LED cells being interfaced with the artificial neuromorphic device for different times. And we could see that the buildup of the dopamine, and the more, let's say, that the cells were exchanging dopamine with the neuromorphic device, the more they would, let's say, keep memory of it. So the synaptic conditioning would be enhanced, as you can see from the conductance of the transistor channel being decreasing largely over time. And also through the microfluidics, we exploited a very, let's say, interesting process here. So when a neurotransmitter is released by cells and goes to the postsynaptic domain, it's not just processed by the cell, it gets being uptaken by the first cell. And so this back and forth of neurotransmitter is actually quite an important point in the neuronal connectivity. And we could emulate this by, again, monitoring the conductance of the neuromorphic device by tuning then the flow rate of the microfluidic channel. And so how much dopamine would be exposed to oxidize and then trigger a conductance change in the neuromorphic device. And then just recently, we have been working on exploiting multiple neurotransmitter modulation of this hybrid synapse. And so we actually focus, other than on dopamine, on serotonin and ascorbic acid, and we have seen exploiting an oxidation reaction. What is the contribution of those neurotransmitters to selectively and individually trigger a different response in the neuromorphic device? And just last, I'd like to show you what is a kind of a typical neuroscience experiment, which we can recall with our hybrid synapse by, let's say, conditioning with multiple neurotransmitters. So we did what is so-called this Pavlov dog conditioning. So the dog, when the bell is ringing, is actually doing nothing. When he sees the food, he starts salivating. And then you associate the bell with the food. And this means, and he will salivate. And then when you remove the food, and you still, ring the bell, the dog keeps salivating, thinking there is food, but it's actually nothing until it goes away. We did the same. So for us, the bell was the dopamine, the food was ascorbic acid and serotonin, and the combination of the two signals was the whole mixture. So we had an increased, a decrease in conductance level, which means for us an excited state. And then we removed the food, and we still had an excited state of our synapse, even if when dopamine was there, and then it would just extinct over time. So this is just to conclude and to wrap up and to say that we really take an effort in trying to design materials for bioelectronics, which, well, they would really recapitulate the functional areas of neuronal tissues, and in particular, what I showed you today was more on organic neuromorphic devices, which are definitely ideal candidates for creating these hybrid and biohybrid interfaces. And in particular, we showed you, I guess, what is the possibility in replicating at least short and long-term potentiation, both with single and multiple neurotransmitters. And yeah, that's a picture of my lab from last year. I also to thank the collaborators which contributed to the work, and I'm very happy to take any question. Thank you.
Thank you, Francesca, for this very nice presentation, and the paper is open for question and discussion if there are any questions. Well, in the meanwhile, that there are questions from the audience, I have two questions myself. One is very much naive. It's about implantation of electronic devices in the human brain. When do you see this is going to be a realistic goal? And if there are some ethical, of course, ethical issues related to this.
Yeah, definitely. I mean, that's a very good question. I've been asked this question since this work came out very often, because obviously the fact that you have a microchip which now can adapt itself could raise, some of these ethical questions. I guess as far as these devices find a medical use to support, to replace, so this kind of device one day could be implantable. This is a very fundamental study. This is done on single cells, but obviously the idea is to have this kind of organic neuromorphic devices to be microprocessors into brain implants to support the information processing on implantable devices. And as far as we can keep it ethical, let's say, from on a medical side, to support or to replace, to restore, not to replace, to restore certain functions, then I guess the technology, I mean, is so we're doing a good job in pushing the technology. But I wouldn't really think of, more like translating this kind of technology towards a kind of a bad cyborg idea. I'm more very much speaking on the medical side.
Okay, thank you. I have a second, maybe less naive and more technical question. Is related to the time scale domain. When I saw your graph, is always on the x-axis is always the time, and it's the time scale it's in the range of seconds, if I understand well. So, what do you think? Is it viable to shift this time scale to lower ranges, more typical of electronic devices, and how this can cope with the biological systems?
That's a very good question. So obviously, the organic neuromorphic devices, they don't want to really replace conventional silicon-based electronics and membristors and hardware, artificial neural networks. So the point is, in this case, what I showed you, those were very big devices. So we have some room for playing in the sense that we can make these devices being faster when we can miniaturize them. So we can create a smaller device itself, to create arrays, and so trying to get close to the silicon technology. However, say it's highly but it's really difficult to get to, let's say, nanosecond kind of switches, right? So this is a polymer. The conductance mechanism is completely different. But moving at least from second to millisecond regime, that would be then even more beneficial for the biological interfacing, because neuronal cells do have action potentials in the millisecond range.
Okay, thank you very much. Are there any further questions? I don't know if you have to raise your hands. I don't know how to handle this technically, but I think you can just switch on your microphone and make your question. No questions? Okay. So, okay, we thank again Francesca. Thank you very much for your contribution and for your answers, and good job, and congratulations for your achievement so far. And so thank you, and then we can then move to the next speaker of this morning session, which is Professor Jing Wang from the technical ETH in Zurich, Switzerland. And his title is Combining Plasmonic Transduction and Photothermal Heating for Direct and Amplified Self-Validating Detection of SARS-CoV-2. Professor Wang, the floor is yours.
So thank you very much, Luca, for the kind introduction. I'm trying to get this presentation up. So, this is okay. So I hope this everyone can see the presentation okay. So the topic I'm trying to give you a presentation on today is on the plasmonic detection. So this technology, we have been developing this technology and trying to apply this technology on the detection of the SARS-CoV-2 virus. So this is the outline. So I think a lot of the audience is probably familiar with plasmonic sensing. So this technology has a lot of different applications. So my field, my specialty is in the air quality. So for the air quality, we know that there are a number of airborne pollutants with biological origins. So for example, nowadays, of course, everyone knows the SARS-CoV-2 virus very well. And in addition to SARS-CoV-2 virus, of course, you have other viruses such as influenza, adenovirus, and also bacteria. And more in general, and also pollen and molds and fungal spores. So there are many different types of bioaerosols. So we try to develop sensors for these bioaerosols. And for example, we have one example. I'll show one example for the total bioaerosol detection. And so once the COVID-19 came, and much of the attention of my group has been shifted to the detection of SARS-CoV-2 virus. I will show you a couple of examples. One of them is a detection, direct detection of the virus. Another one, we use the amplified signal using the enzyme and then do a self-validating detection of the virus. Okay, so the plasmonic sensing, so a little bit of background. So plasmonics, so this phenomena is quite often associated with the noble metals. For example, if you have gold nanoparticles, and if you have incoming light, the electromagnetic field of the incoming light will have interaction with the gold nanostructure, therefore causing a certain signal modulation of the spectrum. So now if this your sensing target, somehow you can attach the target to make your sensing element specific for your target, and then this target can attach onto your gold nanoparticle, for example. Then this will change the local refractive index, therefore change the result from the spectrum, and then you can get a signal for the sensing application. So this is the system we quite often use. So this is the optical detection system. in the common path arrangement. So as you can see, the white light coming in, we have a number of components, the normal one, the collimators, the polarizers, the bifringe crystal, and so in the middle here, you can see the sensing element, so a little bit pink colors, nanoparticle, gold nanoparticles show a little bit pink color here, and from AFM images, as you can see here, the size of the nanoparticles around 40 nanometer, so they form a two-dimensional array on the sensing chip. So this is total internal reflection setup. So for the first application, so we try to detect the total bioaerosol. So the bioaerosol. So in this one, we do not differentiate what are the species, what type of virus or bacteria. So this is just targeting the amine groups. So the amine groups are ubiquitous on a lot of the biological materials. So then this gold nanoparticle is functionalized, intended for capture of the amine group. So once the amine groups come in and then with the reaction, so then this biological component can be attached on the gold nanoparticle, then we can see a signal. So for the applications, we use the environmental materials from the air quality monitoring network in Switzerland. So in Switzerland, we have this air quality monitoring network. As you can see on the map, we have 16 stations. So on these stations, so there are urban stations, traffic sites, and also stations above 1,000 meters above the sea level, so the mountain stations. So from these stations, we have the air samples, and then from these air samples, we can measure their biological components, and we can compare the normal air quality standards, such as particles, the PM, the particulate matter, less than 10 micrometer or less than 2.5 micrometer. So these are the common air quality standards. We also can compare this with biological measurements such as endotoxin. So endotoxin is basically from the cell walls of gram-negative bacteria. So from the measurement, for example, we look at these three different samples from 3 different stations. From these stations, from Bern, Basel, or Rigi Mountain, and we can see here. So the biological components, the amount of biological components are low in the winter, and getting higher in the warm air, in the warm time, in the summer, and in spring, you have higher concentration. So this is correlated to the activities of biological like bacteria. So they are the biological species. So we look at the correlation. So if we try to look at the correlation with bioaerosol and the endotoxin, so the endotoxin and the bioaerosol, so they have quite good agreement. So both of these reach highest concentration in the summer months, and the lower concentration in the winter. But for the correlation with the PM10, this is negative correlation. So the PM10 concentration is higher in the winter. So I think most of people have the experience in the winter seasons. The air quality is worse, mainly because of the inversion layer. So the boundary layer of around the Earth is at a lower position, and the pollutants are not easy to disperse. So therefore, the PM10 particle concentration is higher in the air. However, this is the opposite to the biological activities. So as we can show here, so if you just look at the general air quality standards, so you do not necessarily see the correlation between the general air quality standards and biological materials. So therefore, so our recommendation is that in the future, the improvement of air quality, we could need to look at the more detailed composition of the air pollutants instead of just look at the total mass of the pollutants in the air. Okay, so now I move on to the next part on the detection of SARS-CoV-2 virus. So the SARS-CoV-2 virus, I think by now most of the people have a very clear image of this virus. So this is the schematic of the structure of this virus. So the virus has this is an enveloped RNA virus. So this is the RNA inside, and then on the envelope you have different types of proteins. So these could include the envelope protein, or this spike protein, or N protein. So these different proteins could be used as targets for the sensors. And also they are used, for example, for development of vaccines. So the most widely adopted diagnosis method is the reverse transcription polymerase chain reaction. So the PCR. So the PCR technology. So by now everyone has everyone knows this PCR technology quite well. So the target for PCR detection is the RNA. So first, we need to open this membrane and then release the RNA. And then once the RNA is released, so PCR is basically an amplification procedure. So by using this RNA, the first RNA as the original copy, then go through the transcription, and then from the transcription get to the corresponding RNA structure, DNA structure, and DNA could be magnified. So depending on how many cycles you use, every cycle, then this number of the targets is multiplied by 2. And so then in about half an hour to 1 hour, then these concentrations of RNA can be magnified by millions and millions of times. So then you can get the signal. For example, you have a fluorescence probe, and then you can get the signal from the detection. So PCR by now is basically used as a standard for the clinical diagnosis, but there are certain limitations. This from this thermal, you need to have this thermal control. You cool it down, annealing, extension, and the denaturation. So these steps need different temperatures. To go through this temperature cycle and then control the amplification, we'll need a certain time and need instruments to control the temperatures. So there are a lot of efforts to develop faster and various type of novel sensors for the virus. So my group, since we are working on the plasmonic sensing, so our concept came from plasmonic sensing. So as we already shown, so the plasmonic effect. So once the light coming in from the interaction between the light and the sensing element, you can have different types of signal. So the signal of enhanced electric field. So this one allows you to do direct sensing. So this we already covered in the first example. But there's also another effect. This is called a photothermal effect. So the energy from the light can be converted to a thermal energy to induce increase of the temperature at the local position near the sensing element. So this one is also a well-known phenomenon. So for example, the photothermal therapy, and then you have certain cancer cells in the body, then you could inject certain plasmonic nanoparticles. These plasmonic nanoparticles could be concentrated close to the cancer cell. Now you use a piece of light to increase the temperature close to the cancer cell, then you can kill the cancer cell. So our concept is we can combine this photothermal effect and the sensing effect. So therefore we have a dual functional plasmonic sensing. So not only the transduction of the signal, but also the thermal effect. So this is our system. So our system similar like before. So the light from the left coming in. So this gives us the plasmonic signal for sensing. And then now we have a second piece of light source. So this is the laser coming from the top. This is perpendicular to the first line. This light coming in, this will hit the gold nanoparticles. So this will induce the localized temperature change. So we tune the gold nanoparticles for their nanostructure, and then the wavelengths. So the maximum absorption is happening according to this wavelengths we use. So once we tune this structure, we can get the best efficiency to convert the energy. So here's the characterization of the temperature. So we can the heat up, the local heat up is very fast. So our sensor is not so fast. So we can say it's less than 1 second, but according to theoretical studies, it should be very, very fast, much faster, down below millisecond. So the once you start to heat, the temperature will stabilize, and once you turn off the laser, the cooling down is also very fast, below 1 second. So therefore, we can have a very direct and localized control of the local temperature. So in the sensing is achieved by functionalization of gold nanoparticles with the first we have a thiol group, and the thiol group is linked to the corresponding DNA. So the corresponding DNA, this DNA is manufactured according to the different sections on the gene bank of the virus. So we target different sections of the virus, and then these corresponding DNA, once they see the virus RNA, there will be a hybridization. This hybridization allows capture of the RNA and provides us the signal. So here we look at the temperature control. So if we have the perfect match, then we should have a hybridization. This hybridization, the kinetics of hybridization is enhanced by the temperature. So as we can show here, so this is the curve at the room temperature. This is the curve at the higher temperature when we turn on the laser. So as you can see, so because of the higher temperature, we have a much faster hybridization. So we can improve the hybridization velocity, and also we can improve the lower detection limit. We can achieve a more sensitive sensor. So this is showing the case when we have not a perfect match. This one is not a perfect match. This one is not SARS-CoV-2 virus. This virus is corresponding to the first, the SARS-1. So not SARS-2, but SARS-1 virus. SARS-1 virus is very close to SARS-2, but not a perfect match. So because of this is not a perfect match, when we turn on the temperature, so initially, because of mismatch, they can still hybridize this imperfect hybridization. But once we increase this temperature, this imperfectly matched RNA will be kicked out. So we have dissociation once we increase the temperature. So that shows this can give us a very specific detection. We will only detect SARS-2. We can even differentiate SARS-2 from SARS-1. So here we do the characterization. We try to fix the lower detection limit, and then try to fix that. So we try to do a mixture, not only SARS-CoV-2, but mix this with SARS-1. And then even after the mixture, we can still see a nice signal. So that's our first example. So after this, then we are thinking, okay. So now can we also improve the lower detection limit? So the concept is, can we also do an amplified detection going in a little bit similar in the direction of PCR? So can we amplify this signal? So how do we amplify this signal? So we come up with this concept. So we will use an enzyme-assisted procedure to amplify this signal. So how do we do this? I will give a bit more detail. So the local temperature control, I showed with a higher temperature we have faster kinetics. So first the step. So now we have this sensor. We have 2 steps. So first step is similar like before. We have the corresponding DNA, and then now the target RNA coming in, and then we have hybridization. So this first steps give us the signal. This signal is enhanced by the temperature. So as you can see, the temperature we control from 33 to 40, 41 to 42 to 45. As you can see, when this local temperature is above 41, then we can get stronger signal. So after this step, so after the first step, we have this is on the go nanoparticles, the corresponding DNA hybridized with RNA, and RNA is much longer. So there's still a section of this RNA which is naked and can be used to further application. Then we design a second probe. So this probe is corresponding to this part of the RNA. So this part we design this one side. This probe on one side has the fluorescence probe. On the other side, this has an inhibitor. So when these things are together, then we don't see this fluorescence signal. And then in between, we have an AP site. So this AP site is basically a target for this endonuclease enzyme. So this enzyme will specifically recognize this AP site. So when we do this second step, so we release all these fluorescence probes and enzymes into the sensing solution, and then first we'll have hybridization on this part of the probe. So then the enzyme will recognize this AP site. So this enzyme will cut this AP site. So then this long probe will be cut into two shorter probes. So once they come shorter, then these shorter probes will be released. So once they are released, the inhibitor is far from the fluorescence, and we can see the fluorescence signal. So this is the key here is again the temperature control. So the temperature, because this long probe, entire probe, the melting temperature is at 54. The length is 22 base pairs. So once we cut this, we are down to about 10 base pairs for each shorter probe. Therefore, the melting temperature is lower. So we control the local temperature in between. So the key is to have the temperature about 41. We use 41 in between. So therefore, we can have the hybridization. We can help the hybridization for the long one, but we can release the short probes. So once we have this release of the short probe, and then we can this process allows us to go through a cyclic detection. So the first step we can do this first step, you come in different concentration, we do the calibration for the first step. The second step then, so we can release, so we can continuously put in more enzymes and probes. So every time we cut off some and we'll release more signal, then the signal will linearly increase. Then we can set a certain time. For example, if we set at 800 seconds, then we can measure the signal. So this amplification of the signal allows us more sensing of the lower concentration of the virus. We can achieve trace amount of sensing. So we also do this verification of this sensor compared with clinical samples. And then clinical sample, these are the PCR results. So the PCR positive results, so these four results are clearly PCR results are positive. We also get positive results from our sensor. This result, the negative ones, so these are the patients, so 8 patients results. Negative ones, we also get negative results. So there's the number 5. Number 5 is somewhere in between. For PCR, we need to run the PCR like 40 cycles to get positive results. And then even for this, at the borderline, positive, we can also get a positive result from our second, the amplified sensor. Okay, so I will skip the video. So now we are doing this in the field studies. We get the samples from the hospitals and the nursing homes. So this is direct sampling from the air. And then from the air samples, we sample this in the different nursing locations in the nursing home, and then use our sensor to get the results. Some preliminary results. I will not show the preliminary results. So as a conclusion, so we have a sensor development for COVID-19. The plasmonic sensors has a certain very good features, and we would like to apply this technology to combine the sampling and sensing technology to directly use this in the environmental monitoring and in the combat against COVID-19. So finally, I thank my group and especially Dr. Guangyu Qiu for the very nice work. Thank you very much.
Thank you, Jing, for wrapping up in time, almost in time. First of all, I'd like to apologize for not introducing you appropriately. The background of Professor Wang is the educational background started in China, and then he moved, if I understand well, to Minneapolis, where he did a remarkable job, and then he moved to Switzerland, to Zurich, and his main interest is related to nanoparticles and the impact of nanoparticles, and then on health and safety issues. The paper is open for discussion and for questions. If there are not, I have a question myself anyway. So whoever wants to raise a question, please go ahead. Okay, I'd like to have a more lively audience here anyway. I have a question myself. The question is related to your presentation. And first of all, congratulations, because it's almost 30 years now that people are talking about nanoparticles and nanobiotechnology and the use of nanoparticles in therapy and action and things like that. I think you used a very specific feature of the nanoparticles, which is the plasmonic response. So this is the key feature of nanoparticles that distinguish this system from others. And so this is the very smart way to use nanoparticles in combination with biological stuff. The specific question is the sensitivity of your technique. You show that if you have SARS and SARS-CoV-2, you can detect the differences. So how far can you push your sensitivity? Can you, for example, detect variants of the SARS-CoV-2 or not? So mutations.
Right. Yeah, that's a very, very good question. That's exactly what we are trying to do now. So certainly we are going in this direction. So we believe we can differentiate that. So right now we are doing the test. So we still the problem right now is the PCR has been used very widely, and so we have so I showed like 8 clinical samples. So for real applications, so this is not enough. So in some cases we can see the signal, and sometimes the signal is not perfectly clear. So we have to do more to test more samples to make sure that this signal is really statistically stable. We can see this. So right now I cannot give you a clear answer. We can or cannot, but we are certainly actively doing the verification using a large number of clinical samples.
And related to this, suppose you succeed in the end, in the long run you succeed, maybe in a few months, hopefully. What is the difference in the cost difference in your detection with respect to PCR, and what is the timing as well? I mean,
well, the key idea is the direct detection. We do not need amplification. So because we do not need the amplification, therefore the time could be shorter than PCR. But this the cost is not low. So as you can see here, if we need a laser, we need a spectrometer to differentiate the signal. So the cost is still quite high. At the same time, we are also developing the other types of sensor. So we try to have a suite of sensors. Some of these are more expensive, more looking at more accuracy. Some of these are more faster and cheaper. For example, electrochemical ones. So we are going on both fronts.
Okay, thank you. So in the meanwhile, if someone has some questions, I think there are, and If not, anyway, we have no time anymore, so we thank Jean, Professor Wang again, and thank you, Jean, and it was very interesting. And then we move to the next speaker of this morning session, which is Gianni Ciofani. And if I understand well, Gianni is very much involved. I don't know. Oh yeah, okay, Gianni is there.
Yes, I'm here.
Hi. So you're very much into biointerfaces, right?
Correct.
Yeah. So you're going to talk about your results on this very exciting field. So new materials is interfacing biomaterials, and so this is the very first issue to address. And the title of your talk, Gianni is from ITCMI, if I understand well, Pontedera, and the title of the talk is Hybrid Nanoparticles and Hydrogels for Biomedical Applications. The floor is yours, and then you have how much? 20 minutes. Okay, from now. I'll turn off my camera, and when I switch it on, it means 2 minutes left.
Okay, perfect.
Bye bye.
Thank you very much for the introduction. Good morning to everyone. I start to share my presentation. Can you see and listen to me properly? I suppose yes.
Yes, it's fine.
Okay, perfect. So, as Luca mentioned, I am from Istituto Italiano di Tecnologia, Italian Institute of Technology, and as you most probably know, Italian Institute of Technology has its main quarter in Genova, but then we have several centers spread over all Italy. And more specifically, I am the coordinator of the Center for Materials Interfaces that is located in Pontedera, close to Pisa. At the CMI, I am the principal investigator of the Smart Biointerfaces Research Group. We are a group of about, on average, 15, 20 people, ranging from researcher, postdoc, PhD student, and so on. And our research activities are focused on interfaces and smart nanomaterials. What does it mean, smart nanomaterials? It means that we are interested, of course, in nanomedical application of nanomaterials, but more specifically on the physical properties of these nanomaterials. We would like to exploit nanoparticles not as standard passive nanovectors to deliver drugs or diagnosis agents, but indeed we would like to exploit the smartness of some kind of nanomaterials in order to have real nanotransducers that are able to give also physical stimulation to cells, tissues, and the whole organism. So, for example, we are interested in nanoparticles that are light responsive in order to perform hyperthermia and modulate cell functionalities. We are interested in nanoparticles that are behaving like nanozymes and are responsive to pH values. So, for example, we investigated antioxidant properties of cerium oxide nanoparticles, even in space on board of International Space Station. We are interested in piezoelectric nanoparticles that are able to convert mechanical energy into electrical energy and vice versa in order to stimulate in a wireless and remote mode nerves, muscle, and even cancer cells. And then we are interested, and I am coming to the topic of the talk of today, to hybrid magnetic nanoparticles. We are studying in particular lipid nanovectors that are doped with a magnetic component, most often superparamagnetic iron oxide nanoparticles, and we are exploiting these hybrid lipid magnetic nanovectors to perform hyperthermia and chemotherapy drug delivery to brain cancer. We are especially focused on glioblastoma multiforme that, as most probably you know, is the most aggressive brain tumor with a very, very poor diagnosis. This poor diagnosis is motivated by several factors that are characterizing glioblastoma multiforme, including the topographical diffuse nature that is hindering, in many cases, an accurate surgery, the genetic variability, intratumor, intertumor variability of glioblastoma multiforme, the low specificity of conventional therapies, of chemotherapy drugs, the drug resistance that the glioblastoma multiforme is developing, and last but not the least, the difficulties that many drugs are experiencing in order to cross the blood-brain barrier, that is this physiological and anatomical barrier that is protecting our central nervous system, that unfortunately is hindering also several efficient therapeutic molecules to reach the diseased parenchyma inside the central nervous system. So at the base of our project is the development of a new generation of hybrid lipid magnetic nanovectors that are in principle able to cross the blood-brain barrier and to reach diseased cells in the brain parenchyma. We decided to start from the design of nanostructured lipid carriers. What are nanostructured lipid carriers? Are lipid-based nanovectors, of course, the inner core of which is made of lipids that are liquid at room temperature, most oftenly oleic acid, and this liquid lipid core is protected by a lipid solid shell, most oftenly acetyl palmitate. Inside these nanoparticles, we are loading chemotherapy drugs. In the study that I am showing you today, we have chosen a nuclein-free A, that is a non-genotoxic drug, and super paramagnetic iron oxide nanoparticles. Spions are giving the smartness to our platform, because thanks to spions, we are able, for example, to perform drug targeting by drugging the nanoparticles with the aid of an external static magnetic field. We are able to perform imaging because spions are an excellent negative contrast agent in magnetic resonance imaging. But most importantly, as we will see in the following slides, we are able to perform hyperthermia following a stimulation with alternating magnetic fields. Hyperthermia is particularly important in cancer therapy because it presents a dual effect. Is of course able to induce a direct apoptosis activation in cancer cells, and it is well known that cancer cells are more sensitive with respect to healthy cells to increment of temperature, but also it is able to improve the effects of chemotherapy drugs, because cancer cells following the localized increment of temperature are becoming more sensitive to the chemotherapy drugs. And this is extremely useful because allows us to keep low the administration, the concentration of the chemotherapy drug, and will allow us to hinder the insurgence of chemotherapy drug resistant in cancer cells. The nanoparticles are moreover surface functionalized, and in this specific example, we have chosen AngioPep2, that is a short peptide that has a dual targeting functionality. It is able to promote the blood-brain barrier crossing because the receptor for AngioPep2 is overexpressed on the endothelial cells of the blood-brain barrier, but indeed it helps also the selective uptake by cancer cells, because even glioma cells are overexpressing the receptor for the AngioPep2. So this functionalization, as we will see, is able to perform a dual task in our system. This is a bit of characterization of the nanoparticles that we have developed. You can see some representative transmission electron microscopic images, evaluation of the stability. They present a good stability in different environments and also long-term stability, presenting a satisfactory zeta potential. Functionalization, we achieved a quite good functionalization in terms of surface density of this short peptide, and we achieved also satisfactory drug payload with drug encapsulation approximately of 4-5%. The first step is, of course, the assessment of the selectivity of these nanovectors. In order to efficiently and reliable assess the selective targeting of these nanoparticles, we performed experiments by setting up complex in vitro models that are based on multicultural dynamic bioreactors. Here you can see a photo of these bioreactors during a typical experiment and the schematization. As you can see, the bioreactor is connected to a peristaltic pump that is allowing us to mimic the shear stress values that we are naturally experiencing in the brain microcapillaries. And the bioreactor is composed by several chambers. Each of them can be cultured with different cell lines in order to comparatively assess the uptake of the nanoparticles. In this experiment, for example, we exploited differentiated neurons, glioma cells, U87MG, healthy human astrocytes, and healthy human endothelial cells. The nanoparticles are injected in the fluidic system, and then the selective uptake is evaluated. As you can see from the qualitative 3D confocal reconstructions and from the quantitative evaluation of the uptake, the targeting abilities of the nanoparticles are excellent towards cancer cells with respect to all the other healthy cell lines. As you can see in this plot that is summarizing the results. Extremely important, as you can realize from this data, is the functionalization with the AngioPep2 on the surface of the nanoparticles. Just in this case, we can achieve an excellent selective internalization by cancer cells. So, as I mentioned, not only the targeting is important, but also the crossing of the blood-brain barrier. Also, in this case, we established a quite complex in vitro model of the blood-brain barrier with this dynamic bioreactor that comprise two chambers, one static chamber where cancer cells are cultured, and a fluidic dynamic chamber where the culture medium can flow, simulating the blood flow. These two chambers are separated by a porous membrane, and this membrane is mimicking the blood-brain barrier. In fact, this membrane is co-cultured with human astrocytes and human endothelial cells. When the properties of the blood-brain barrier have been verified in terms of transendothelial resistance and in terms of permeability of typical tracers such as dextrans, we start the experiment by injecting the nanoparticles inside the system, and we evaluate the blood-brain barrier capability of the nanoparticles. Again, as you can see from the 3D images reconstructed through confocal acquisitions and by the quantitative evaluation of the BBB crossing, the functionalization of the nanoparticles with AngioPep2 is fundamental in order to promote the crossing of the blood-brain barrier. So after the assessment of the blood-brain barrier crossing and the selective targeting by glioma cells, we started to investigate the therapeutic efficiency of these nanoparticles. As I said, these nanoparticles are attacking glioma cells by both exploiting chemotherapy provided by the nuclein-free A that is loaded inside the nanovectors, but also thanks to the hyperthermia that is developed following the stimulation with the alternating magnetic field, and thanks to the presence of the spions inside the lipid matrix of these nanovectors. We investigated quite carefully the mechanisms at the base of the efficiency of the hyperthermia that developed following the electromagnetic field stimulation. And we realized that the therapeutic effects that are following upon this hyperthermia treatment are due to the disruption of the lysosomes where the nanoparticles are accumulated. This is particularly clear from this video that I am going to show you if it will work, where lysosomes are stained in red, the lipid magnetic nanovectors are stained in green. We realize that the colocalization of the nanovectors with the lysosome, and upon the stimulation of, thanks to the alternating magnetic fields, as you can realize from this video, the signal of the lysosomes is disappearing. I would like to show you again if it works. So it means that we have a dissolution of the stabilization of the lysosomal membrane, and the lysosomes are spreading all their digestive content in the cytoplasm of the cancer cells. As you can see, we immunostained the catepsin B, that is one of the main enzymes that is present inside the lysosomes, and following the stimulation with the magnetic fields, the catepsin B is spread in all the cytoplasm of the cells, contributing to the therapeutic effects that we are going to see shortly. Just a quick note about the efficiency of the drug that we exploited, and the efficiency of nuclein-free A is even enhanced when it's delivered by the nanoparticles because of the hydrophobic nature of this drug and the lipided nature of the nanoparticles. So we have an improved internalization of the drug by the cells, but most importantly, the effects that are stemming from the combined therapy originating from chemotherapy and hyperthermia. As you can see, both immediately after the chronic stimulation with alternating magnetic field and after 3 days since the end of the chronic stimulation, we have a dramatic decrement of the cell viability in the combined experiment chemotherapy plus lipid magnetic nanovectors. As you can see, the cell viability is decreasing almost to zero. As I mentioned before, these are other results that I am not going to describe because lack of time. I think I have just a couple of minutes. As I mentioned before, we investigated the effects of hyperthermia in a more precise way, and we evaluated the activation of the apoptotic pathways, and in particular, we would highlight as following the stabilization of the lysosomal membrane and the spreading of the digestive enzymes in the cytoplasm, the caspase 9 pathway is activated and the cells are undergoing quickly apoptosis. So these are the main conclusions. We have developed a new kind of nanovectors based on a lipid matrix loaded with the superparamagnetic iron oxide nanoparticles and the nuclein-free A as a chemotherapy drug. We demonstrated that upon functionalization with angiopep2, these nanovectors are able to cross and complex in vitro models of the blood-brain barrier and to efficiently reach an in vitro model of glioma. We highlighted the pathway of apoptosis activation after hyperthermia treatment, and now in these months we are performing in vivo experiments following animal injections with these kind of nanoparticles. So thank you very much for your attention, and I'm available for any question you may have. Thank you.
Thank you, Gianni, for this very interesting presentation. And again, also to you, apologies for not having introduced you appropriately, but maybe it's nice to introduce afterwards. And the background from Gianni is graduation at the University of Pisa as a biomedical engineer, something like that, and then he got a degree at Scuola Superiore Sant'Anna di Pisa, which is one of the excellent centers for this kind of studies in Italy. And then he moved to IIT, and he's at the same time, if I understand well, professor at the Polytechnic Institute in University in Turin, and he has a tenure in IIT.
Yes, right? I have been in the past, almost 5 years, associate professor there.
Okay,
yes.
So you moved back to IIT.
IIT, exactly.
Well, okay, this is just to warm up some, questions from the audience, which is maybe, I see it's a very good audience in terms of number, but not that. Oh, there's Giuseppe. I see yes. Giuseppe Strangi, a question from. So there are many questions now. Yeah. So
may I start?
Yeah.
Okay. First of all, thank you very much, Gianni.
You're welcome.
I think that your talk is very nice, and I have a question regarding the blood-brain barrier and the way in which you are mimicking, this membrane. The idea is great, but you mentioned that the functionalization of the particles is key, is crucial. Can you please comment on that?
Yes, it's crucial because, as I think most probably you know, the blood-brain barrier is a very selective component of the central nervous system, so it is hindering the passage of most of the molecules and nanoparticles. These nanoparticles present a low tendency to cross the blood-brain barrier because of their lipid nature, but it would be not enough in order to achieve a high concentration in the brain parenchyma. So the functionalization is crucial because angiopep2 is able to target some receptors that are expressed on the surface of the endothelial cells, and following the combination targeting receptor angiopep2, the nanoparticles are undergoing a transcytosis. So they are crossing the endothelial layer of the blood-brain barrier and are exocited in the parenchymal compartment. Without this functionalization, the amount of nanoparticles that would reach the parenchymal compartment of this blood-brain barrier model would be extremely low.
Thank you.
Welcome.
Okay, then we have a question from Luisa De Cola, University of Milan.
Yes, thank you very much for your beautiful talk. I have a question about the functionalization to allow to cross the blood-brain barrier. I did not understand what is really the peptides or the system that would allow the crossing of the blood-brain barrier. I understand the targeting units very well, but I could not cut what allows the lipids layer to cross it. So maybe repeat it, sorry.
Yeah, no, yes, you are correct. Maybe I didn't mention because I was a bit running. So on the surface of the nanoparticles, we have a layer of the SP PEG. So there is a double functionalities of stabilizing the nanoparticles in aqueous environment, but the PEG presents some reactive groups that can react with the angiopep2. So we can perform a functionalization by simply covalently attaching the angiopep2 to the functionalities of the SP PEG by exploiting NHS chemistry very simply.
And the zeta potential is very negative. So where does negativity come from? From the peptide that is highly negative, or?
It is coming from the peptide, yes, after the functionalization with the peptide. We have a quite a high density of a peptide on the surface, and we have a shift towards negative zeta potential following the functionalization, correct?
Okay, thank you so much.
You're welcome.
Okay, we have no time left, but I'm still curious. I have a quick question myself, which is related to a very quick question, which is related to the size of this lipid matrix. I don't know if it is a lipid bilayer or if it is a lipid matrix, and if you can control the size, and if yes, what are the effects that you, if you study the effect of the size effect of this matrix?
Yes, it is not so simple to control the size. Let's say that our idea is to try to obtain nanoparticles in the range of 100 nanometers, even low is possible. But it's not so easy to control the size. Unfortunately not. With the procedures that we are exploiting in order to fabricate these nanovectors is a sonication followed by hot temperature homogenization. And of course, by changing some parameters, we are able to tune the size, but it is not so much a reproducible process. What we made is an optimization of the fabrication process in terms of temperature, pressure, and so on, in order to have nanoparticles that are as small as possible.
Okay, thank you. So I think we have run out of time, so we move. We thank again Gianni
Thank
for
you.
this very interesting and for finally triggering some question from the audience. And then, okay, we move to the last speaker of this first batch of this morning session. I'm honored to have Luisa dell'Aquila from University of Milan. And I will introduce her as it has become a tradition for this morning session afterwards, after the presentation. So the title, go ahead, the title of the presentation is Hybrid Nanoparticles and Hydrogels for Biomedical Applications. Luisa, the floor is yours.
Good morning. I hope you can hear me and see my screen. Luca, can you confirm that everything is okay?
Yeah, everything is okay, so please go ahead.
Okay, so thank you for this beautiful meeting, and thank you for inviting me for this keynote lecture. So what I would like to share with you today is a little story that has to deal a little bit with nanoparticles. We heard a lot this morning already on nanoparticles, but in particular, I would like to point out a new type of particles that we make in our labs that are called nanocages. And I will explain to you in a moment what they are and why they are special for us. And then I would like to show you that this nanoparticle in general, and in particular our particle, can be a component of a much more complex structure that are these hybrid hydrogels that in our case are not scaffold in the sense that we do not make them in the lab and then in plant, but we form them directly in situ in the tissue where we want to have some kind of healing or function. And I will show you some in vivo application already on large animals, so some of these hydrogels, just to give you a feeling how these materials behave once they are injected into the body. So let me start with a very, very short introduction, just one slide about nanoparticles. We heard a lot about these beautiful systems that can be stimulus responsive, or they can be of different material like liposomes, already we heard a lot. They can be polymeric or hydrogel. In our case and in our lab, we mostly focus on mesoporous silica nanoparticle. So these are micelle template silica system, where of course we can tune the porosity, we can tune the size and the morphology. I will show you one example. Or we also work on nanocapsules, and in this case they are also silica based, but you will see in a moment that our silica is not 100% silica because it contains some groups that give some kind of functionality but also breakability of this particle. And indeed, when we started this work on nanoparticles, we were a little bit confused by the literature because the silica was a little bit schizophrenic in terms of behavior. So if you look indeed, you will find out data on silica degradation of a few hours or a few weeks or even months. And so we decided to go for a new approach where we could control this degradation by an external stimulus. And so we design, as you can see here in this simple cartoon, particles where we could introduce into the silica framework some kind of organic groups. And these organic groups are very special because, as you can see on the formula on the right side, they can be groups that are indeed covalent reversible groups, like the disulfide or the ester or the imines. So that means that, for instance, if I take the disulfide, I can reduce the disulfide to thiols. And doing this, of course, I will break the SS bond in order to form two thiols. But during this break of this bond, you can imagine that you break then the silica because you will break the particle themselves. And therefore, you will end up with particles that do not degrade, but break in small, tiny pieces that can be excreted by the body of the animal. So we work quite a bit on the groups, and just to give you an example, we work with peptides that can be enzymatically cleavable, or we work with imines that can be pH responsive, and disulfide that indeed are responsive to natural reducing agent like glutathione that is present in any type of cells. Of course, this responsivity can also be of a physical type, so you could imagine, for instance, groups that are light breakable or thermal breakable or ultrasounds breakable, and therefore you can shift the attention from chemistry to physics in terms of input. But let me tell you one example of this particle, just to give you a feeling of this breakability. So this is an example of mesoporous silica nanoparticle, 100 nanometer in dimension, where you can see by TM very clearly that already after 24 hours, the particles start to be kind of broken, they lose the morphology, and then you see the formation of these big holes that eventually result in a completely destruction of the particle in tiny fragments that are between 3 and 4 nanometers. So we studied this process in detail, not only in vitro, but also in vivo. And we found out indeed that after already 4 days, all this silica material is present in the urines of the animals. So following this approach, we decided to look at the effect of the morphology of this particle. So we produced a series of nanoparticles that, as you can see from these snapshots, they can have a different morphology and different even chirality. So you see here a kind of summary of our nanoparticle that can be disc shape or elongated rods with a chirality that can be induced by the use of a chiral template when we make the micelles, or we can make flat hexagons or these funny looking what we call UFO objects. So some of these particles is under investigation to understand the different biodistribution due to the shape rather than on the size. But let me show you also that we can shrink the size. And as already commented, this shrinking of the size could be important because biodistribution, of course, depends not only on the shape, but also on the size of the particles. And doing that, we realized very soon that slightly changing the condition to make this breakable nanoparticle, we could obtain two different morphology that you see here in the two different TM images. Perhaps you recognize that in the TM in the bottom, you can really see the mesoporosity. So you can really see that the particles are mesoporous. You can even see the channels of these 20 nanometer particles. But on the top, perhaps you cannot recognize indeed the morphology. And despite the fact that the particles are extremely monodispersed, and they have a very interesting distribution, it took us some time, and in the meantime, I have to say, we were stood by Ulrich Wiesner, who published on a silica-based nanoparticle very similar to ours. And indeed, looking at TM, we found out that these particles have a very special feature. They have essentially a single hole, but they have different entrants, and the entrants are between 3 and 4 nanometers, while the space around this, what we call nanocage, is about 10 nanometers. So this is quite an unusual morphology for this material, and you will see in a moment that because of that, we have a very unusual cell internalization of this material. So, of course, this particle can be easily functionalized. So we can imagine, for instance, to attach, to visualize the particle, an imaging agent like cyanine 5, that we can use for optical imaging. And you can also see from the XPS that we can determine the amount of disulfide groups present into the structure of the silica. And of course, you can calculate by TGA eventually the loading of the material inside or of the cyanine that is covalently linked on the surface. This is just to give you a feeling of our characterization means, but also very important for us was to characterize the degradation time of this material. And here you see that the particle that have been indeed treated with glutathione, and this is a 9 millimolar concentration of glutathione PBS, so very similar to tumor cell. Essentially, the particle already after 12 hours are gone. So we lose the morphology and we find into the DLS only very, very tiny fragments. Also, of course, this fragmentation is accompanied by the release of molecules that can be trapped inside. As you can see here, for instance, with the example of the release of a Earth molecule. And Earth is an interesting molecule because not only is released, but also it migrates into the nucleus of the cell. So we can look at the kinetics of the release and migration inside the cell. And now you see that indeed the particles are very stable if we do not add any glutathione, but they are quite quickly degraded when the glutathione is added. It takes about 6 hours. So with all these things in mind, we started to look at the biodistribution of these particles inside the mice. And this came as the very first surprise because we functionalize this particle with glucose in order also for us to look at the uptake into the brain, even though this is an IV administration of this particle. And indeed, we see that about 1.3% of the administered particle go indeed into the brain. But with our great pleasure, we see that most of the particles go into the lungs. And this is a very interesting result, especially now with the COVID, because it's very hard to target lungs. And so for us, it's an interesting result. And of course, as you could imagine, the particle end up into the liver, in the kidney, and in the spleen. So this is a quite normal biodistribution for this type of size of particles. So taking into the advantage that they go to the liver, we then look at the toxicity of this particle, also in the different organs. So this is an acute toxicity. So after 24 hours injection, we sacrifice the mice, and then we look indeed at the immunoresponse that this particle can create. And as you can see from this very quick histological analysis, you see that none of the particles create an immunoresponse. And this is not surprising because, as you might know, silica has been FDA approved in 2011, and also the SS group that we use are FDA approved in the community. So this is quite a nice result. So this means we can go in vivo. And to go in vivo now, we team up with a group of Giovanni Sitia, the San Raffaele in Milano. And what we did, we xenographed a liver tumor into the mice in order to look indeed at the distribution of this particle into the tumor and eventually if this particle would be able to reach the tumor. We also look at the effect of the presence of the sugar on the surface in order to see if this would improve the uptake into different types of cells. So we administrate the particle again intravenously, and then we look at what happens after administration in these mice. And now you see essentially what happens that is quite spectacular because you see that in red we have the cyanine labeled nanoparticles, and in green we have the macrophage system, and in white essentially you have endothelial cells. And what you see is quite unusual because normally we would expect the nanoparticle to be eaten by the macrophage, like often happens with the silica nanoparticles. But in this case, we don't see any presence of the particle into the macrophage, while we see a huge presence of this nanoparticle into the endothelial cell as well into the tumor cell. So we repeated this experiment in vivo and in vitro several times because we could not believe these results. It was quite unusual. And again, we see that the particle clearly enter into the endothelial cell while they do not enter into the macrophage. And we repeat these results also in comparing this nanocage with silica nanoparticle of exactly the same size and the same functionalization. And we prove again, but this was already known in the literature, the 20 nanometer silica nanoparticle are indeed internalized into the macrophage. So this macrophage escape is something unique for this morphology and also for this chemical composition of the system. So of course we are going on at this point because we believe that since we can reach the tumoral area, that we can at this point release a drug much more efficiently than with any other type of nanoparticle that are indeed filtered by the macrophage. So with this nanoparticle and the other type of nanoparticle, we decided also to construct a more complex system, like I was mentioning to you in the beginning, into the introduction. And so we decided essentially to use the nanoparticle as cross-linking points in order to form hydrogels. I think everybody knows that hydrogels are 3D network, and in our case, these are covalent polymers, and that can entrap a huge amount of water. In particular, typically we can entrap up to 95 or 98% of water, leading to a material that is essentially made of water and only in a couple of percent made of nanoparticle and the organic link. So this was quite interesting for us, and we decided to go for this type of hydrogel, and in particular, I will show you some of them. And of course, you know now what type of nanoparticle we are going to use in breakable system that can release drugs or any other biomolecules. So what type of hydrogels can we use? And so when we started this work, we were very ambitious because we want some kind of injectable hydrogels. So we want to form the hydrogels not on the bench and then to insert them as a scaffold, but we want to form them directly into the tissue. So we want something essentially that would be injectable as a liquid and solidify in few seconds or minutes into the body. And so, of course, the requirements are quite stringent because we want the reaction proceed in water or physiological exposed media. We want that the reaction works at body temperature. We cannot warm up the body. And we want, of course, the reaction is quantitative and fast. And we don't want any catalyst. Well, of course, we don't want any chemical catalyst that could interfere with the toxicity of the system. And we cannot use light or other catalysts that normally are used. So with this in mind, we look at the literature and we realize that one good reaction is the Michael addition reaction that was explored in the past by an Italian group, that is Ferruti, to make polymers. And indeed, this reaction works very, very nicely in water at room temperature. It's a typical reaction between amino groups or thiol groups and acrylates. And this is so versatile that essentially you can place the acrylate on any material you wish, and you can use any amino or thiol groups in order to create this polymeric system. And of course, in our mind, if we work with porous capsules where we can entrap either nutrients for the cell or drugs, we could also use the particle as a cross-linking agent because they will behave like a huge dendrimers, since many, many hundreds of groups can be linked on the surface of this particle. And therefore, the amino or the thiol can react with the acrylate that is present. So we show that we can use this particle indeed and this material for instance, mimicking chemotaxis, so the recruitment of stem cell thanks to the release of chemokine from the nanoparticle. But we also show that these materials are quite unique because the solidification occurs in a few minutes. And as you can see here, they lead to a completely transparent hydrogel that is very mechanically very stable, as you can see from this upside down vial test tube test. And what is more important for us, the material is extremely porous, as you can see here in this beautiful BioSCM image. Also, in our case, this material is about 93% of water, and again, this proves this kind of water material that we are able to make, even though it's a solid. So we look at the biocompatibility of different types of gels based on this Michael addition reaction, and we saw that the metabolism of the cell is not influenced by the presence of the gel. So the cell proliferate and move into the gel in all the directions. At this point, we were aiming to what I mentioned to you at the beginning. That was this ambitious idea that we can somehow inject directly the solution and then look at the gel formation directly into the tissue. And now you see indeed the end of a surgeon that is prelevating with a syringe our pre-gel. So you see that the solution is completely liquid, so we can use an insulin needle. And in this case, we color the gel in blue, but the gel will be transparent, just to make it visible. And I'm sorry for the next couple of slides that perhaps are quite cruel, but I want to show the real experiments of the surgeon on the animal. And so I will show you already the big animals experiments, the one that have been published, in order to see indeed what is the effect of this pre-gel solution into the tissue. And now you see that the syringe is penetrating into the submucous of the stomach of this animal. And you notice already that where the gel is essentially injected, it does not diffuse into the tissue. So it is essentially placed and it stays in the place where the injection occurred. Normally, a solution would diffuse into the tissue very rapidly, but this was not the case. And so we repeat this experiment, and then we cut essentially the tissue in order to understand what was going on. And now you have the answer where the diffusion cannot occur, because the solidification of the gel, so the formation of the gel, is indeed so fast that after injection is almost immediately formed. And what is even more important for us is that this type of gels are extremely adhesive to the tissue. So you can see that the tweezers of the surgeon are not able to detach the gel from the skin, even though it's pulling the skin of the pig. So we started a long collaboration with the hospital in Strasbourg, where I was before moving to Milano. And we indeed look at the use of this material for tumor resection, but there is a published work, so you can look at the literature if you are interested. But also we look more recently to an unmet clinical need that is something extremely important. That is unfortunately not yet a solution that is essentially to cure or to resolve the fistula that are formed either by a chronic disease or by resection of tumors or other disease. And so just to make a long story short, a fistula is essentially either a hole into an organ, so it can be the intestine, the stomach, or a connection between two hollow space, for instance, between an organ and a vein, or between two parts of the intestine. And why this is an unmet clinical need? It is because, unfortunately, we do not have a material that is able to close this hole or to avoid this connection. So what happens at the moment is that the surgeon has to essentially stitch this hole in order to avoid the gastrointestinal fluid or other, of course, fluid can percolate and can even destroy the intestine. So this is a big problem, and there has been many attempts to have a type of glues like the cyanoacrylate that you can imagine as a chemist is an extremely toxic material in order to close the fistula. And the big problem of the fistula is indeed the fact that it's an open wound. So essentially, if you would inject a solution, the solution would percolate on the other side, except if this solution is able to gelate so fast that would close completely the hole or the tract between the two parts of the intestine. And so we thought that some of our hydrogel, and in this case we use hyaluronic-based hydrogel, would be able to do this. And in order to do indeed this experiment, we had to create a model in large animals that are as similar as possible to human beings. And these are indeed pigs where they do not suffer of fistula, but where the fistula can be created, essentially just introduce a cannula, for instance, in the esophagus, and then remove the cannula after 21 days. So in this case, where the cannula was, you have a fistula. So you have this tract of skin that is empty, of course, in this case. So you have a big hole between the esophagus and the external part of the animal. And so we used this as a real model, and what we did was, together with the hospital Policlinico Gemelli in Rome, was to have as an internal control a second fistula on the left side of the animal. So in any animals, essentially with the right and the left fistula, so to have an internal control in order to have the best control that could tell us what's happening once we fill the material. And so the last few slides, I want to share with you the results, and I want to show you the formulation that we have ended up to do with this hyaluronic acid. So you see here that the hyaluronic acid we use contains 98% of water, and it has a mechanical properties that is 6,000, in this case, Pascal. And this is essentially for us important because we reproduce the mechanical properties of the esophagus. So at this point, we inject this pre-gel, so the solution, into the fistula, and then we left the animal into the farms for 2 weeks in order to check the cicatrization, so the reformation of the skin into the fistula. And now you see the results in these last 2 slides, where you see very nicely that on the top side, in the B and in the right part of the A, you see that the fistula has been completely closed, so the skin has been reformed thanks to the action of the gel, while on the left side of the A or in the C and the D, the fistula unfortunately is open. And this is even more clear if you look not endoscopically, but if you look outside the animal, where you clearly see that after 2 weeks, the wound is still open, it's even infected because the animal was free to run into the farm, while the wound treated with the gel, it forms this kind of pinkish new skin, indicating that the skin has been reformed and the fistula has been sealed. So with this, I would like also to show you one slide on the histology, and you see the formation of this giant cell that correspond to the cicatrization. So I would like to conclude, and I hope that I could convince you that we can design some kind of material for in vitro and in vivo application. I would like to thank the people that contributed to this work, that is my group, and the hospitals that have done all the in vivo works together with us, and of course the funding agents, and all of you for your kind attention. Thank you.
Thank you, Luisa, for this very interesting talk. I would like to remind the audience that Luisa, she gave outstanding contributions in the field of materials for health. She published more than 380 papers with an H index of 77, and she is professor at the University of Milan, and she's director for the Materials for Health laboratory at the Mario Negri, if I understand well. And she is also honorary professor at the University of Tianjin in China. This to warm up the audience. If there are any questions, please raise your hand. There's a time for 1 or 2 questions, 1 or 2 big questions. If not, I have a question myself. I was my interest was triggered by your nanocage particles.
Yes.
Okay, so I have a general question, a very naive question about these nanoparticles, and nanoparticles are used to address tumor diseases, and you showed a slide where the uptake of the nanoparticles is not homogeneous in the body, but it happens to be different in different organs. Okay, so the very question I have myself since years is there a rationale for this uptake? You just let the nanoparticles go through the body, and then the uptake is different as the organs are different, but how do you decide what is the shape of the nanoparticle to address a specific organ? Or so what is the rationale behind this, the uptake?
So unfortunately, there is no real rationale, but there has been a review by Mauro Ferrari a few years ago in Nature Nanotechnology, where he explored indeed both the size and the shape distribution into the body. And so what it shows is that there are, of course, size-dependent biodistribution. So typically, particles above 100 nanometer are accumulated into the kidneys, into the liver, and particles that are typically smaller than 50 nanometer goes to the lung and the spleen. And of course, for the brain, as we heard also before, we must be much, much smaller, hopefully smaller than 20 or 30 nanometer in order to penetrate the blood-brain barrier. So there is a size dependence, and there are several beautiful papers on the shape dependent, because the way these particles tumble and interact also with the cells depends on the addition of the surface of the particle on the membrane. And so as you can easily interpret, particles that are very flat, they could enter not with the flat part, but more with the vertical part in order to penetrate even faster than, for instance, a spherical particle. This is something we also show in a paper. And so the size and shape is somewhat understood. However, one has to be very careful because, like I said, we compare the silica nanoparticle 20 nanometer with our cage that are also 20 nanometer. They are both more or less spherical, but they behave completely different. And here we don't have a rational explanation. What we think, however, is that the introduction of the breakable group into the silica reduce the negative charge of the silica and increase the hydrophobicity because the silica is hydrophilic. And so we end up with a particle that is a kind of schizophrenic, if you like, is a dual behavior, is hydrophobic, hydrophilic, and is negative and positive charge simultaneously. So it's a very funny situation where they resemble a little bit the work of Francesco Stellacci on his gold nanoparticle that are patchy system. And so this is what we believe, and this is why we think that this escape from the macrophage can be triggered or chemically and not only by the morphology of the particles. So I hope I was able to answer your question.
Yeah, and this leads to my second more technical question, which is about these cage nanoparticles, and there are holes of the size of nanometers. So it's a question and a comment. I wonder if water in this case and the presence of water has to play a role. It's confined water, so I wonder if there are some supporting studies of dynamical simulation of
Yes, there is some work on indeed in water in zeolites and mesoporous material. So indeed the water does not behave as a water. This is also somehow true in the gels. And this is why you have a solid material, even though 98% is water. So it behaves more like, I would say, ice. So the hydrogen bonds are highly promoted. So the water in this cavity is not really water in the sense of fluids that we imagine. I have to comment on this because thank you for the question, because indeed the fact that we have this open space is quite unique because on one hand the particles are accessible from the outside, but on the other hand the particles are able to stabilize species that would not exist in solution. So this is a paper we just published in JACS, if somebody is interested, to show that we can capture essentially out of equilibrium species and stabilize them into this cage and eventually release them again into the cell. So it's not only a way to transport a molecule that would be, let's say, impermeable to cell, but it's also a way to entrap a very maybe reactive species that can be then released inside the cells when they reach the target. So this is something just for inspiration, maybe somebody can think about it.
Thank you, very much interesting. Okay, if there are no further questions, I see no raised hands. So we thank Luisa again for this very interesting talk. And okay, so there's time for a very short break. I think we can convene at 11:28 and we start again at 11:30. So coffee break. So we're ready to start for the second batch of this morning session on advanced materials for nanobiotechnology. And so with no further hesitation, I would like to invite to the floor Giuseppe Strangi from the Case Western University, USA. And the title of his contribution would be layered metamaterials from hyperbolic dispersion metasurfaces to fan resonances optical coatings. Please, Giuseppe, the floor is yours.
So thank you very much. Can you hear me?
Yeah, it's
Okay.
fine.
I think that you don't see my presentation, right?
Not yet.
I don't know why.
We see a black screen with a PowerPoint, and here is
What about now?
presentation, and it's not in full screen yet.
I'm trying to go to presentation mode.
The full screen.
Is it in full screen now?
There you are. Okay.
Okay. So first of all, thank you very much, Mr. Chairman. And I would like also to thank, the organizers of this meeting for inviting me. This is a pleasure and an honor to be here. And today I would like to talk about what we are doing in the framework of layered metamaterials, and in particular about hyperbolic dispersion and how we apply this to nanomaterials to try to provide a solution for biotech. And let me say that I would like to start acknowledging, my research team and my collaborators. Nicole Steinmetz at the University of California in San Diego. She's a virologist. Nima Sharifi at the Cleveland Clinic is an oncologist. Francesco De Angelis of the Italian Institute of Technology, Plasmonics and Nanotechnologies, and Federico Capasso at Harvard, working on flood optics. And the bunch of talented students that, without them, nothing of all that I'm presenting today would be possible. So the framework in which we work is theranostics, and we try to use, nanostructures that can have some specific applications. In particular, if I take a pointer, you should see my pointer now. We have at the center of this, we have a multifunctional nanostructures which behaves as an antenna, but these are cell-specific targeting nanostructures that are biocompatible, and so that they can circulate for a long time. They can stimulate a specific immune response, and we know now with the vaccine what is the meaning of stimulating in a specific immune response. They can do controlled drug release. They can increase locally the temperature, or can do multiplex detection, imaging, and sensing. So in this framework, if what we can exactly do here is do, for example, some cellular exploration by using, for example, plasmonic nanoparticles. So these plasmonic nanoparticles can, as you see here, for example, can be used to open or close ion channel, so the control of ion channel aperture, cellular targeting and imaging. They can do photothermal therapy by illuminating these particles when they are internalized via endocytosis. You can shine light, increase the temperature locally, induce some apoptosis or damages to the cell. You can do microRNA detection, Cyrus, molecular imaging via PRET, gene delivery, and many, many more applications and many more things. So this is a real, revolution, which we can go inside the cell, and by using these particles, we can have many functionalities activated by light or ultrasounds or any other electromagnetic radiation. So now the materials, because this is a materials conference, the material that we use are metamaterials. And though this word is abused, for us, metamaterial is nothing else than something that you design and create artificially to have some specific functionalities that can be used for therapy, for diagnostics, for monitoring, for controlling, and so on and so forth in this specific field. So now what I would like to present to you today is some metamaterials for precision medicine. In particular, on the left side, you see this plasmonic nanostructures. Typical one, these are core shell, gold core, or noble metal core, silica shell, and then you embed in this silica shell some, drugs, so fluorophores, whatever you need in order to make some, studies or activate some properties inside your system. Or you can use, for example, hyperbolic metamaterials for sensing and imaging. And on the right, there are a multi-layer stack of gold or metal and dielectric in which you have this kind of a hyperbolic dispersion in use. But also in collaboration with Nicole Steinmetz at University of California in San Diego, we are working on making viruses plasmonic. And to do that, we use tobacco mosaic virus that are decorated with gold nanoparticles. This is an illustration of it, but you see the SEM image of these nanoparticles on top of this plant virus is a tobacco mosaic virus. And we use this for drug delivery, intracellular imaging, or in situ cancer therapies. Well, the question is, what is an hyperbolic medium? Because we are talking about this hyperbolic dispersion in the title. Tell me what's that? Well, the point is the following. If you take an isotropic medium, we all know that the way in which you describe the field in the medium is through this beautiful expression which comes directly from Maxwell's equation. Well, now this, the permittivity here is, in the case of isotropic medium, is a scalar function. So the dispersion equation has this kind of relation where in the K space, this is the isofrequency, and this is the isofrequency surface is a sphere, and we all know that. What happens when we have an isotropic medium? Well, the permittivity is not anymore a scalar function. We need a tensor, and when you have a tensor, you have the diagonal of the tensor, and the elements of the diagonal have a lot of meaning. And the meaning of that if it's all positive, you have a dielectric. If it's all negative, we have a metal. What if you have, something which is not a metal or not a dielectric, in all the directions? Well, if in all the directions you have a metal or a dielectric, you go write the dispersion equation, and you will see that this is the isofrequency surface is an ellipsoid. But if you flip the sign of one of these three, for example, EZZ, and is negative and the other two are positive, it means that in the plane is a dielectric and across it is a metal. Well, what happens is that still you have a scalar, still you have a tensor, but this tensor gives you the dispersion equation, which is an hyperbola. So this hyperboloid is the isofrequency surface in K space, and it opens a huge, enormous space of opportunities. And I can summarize this in, first of all, in the infinite density of states. If you calculate the density of states, electronic and photonic density of states, well, what happens there is that the isofrequency surface is not a bounded surface, so you will end up with infinite density of states. Well, what's the consequence of that? Well, it means that you can access large wave vector modes, and so you can increase the field confinement all the way down to a single nanometer. You localize the field, concentrate the field, and you can have a very intense local field. On the other side, you can have polaritons, and these polaritons propagate with a direction-dependent wavelength, which means that now your wavefront is not convex anymore, but is concave. And concave wavefront gives anomalous interference phenomena that you can exploit in light-matter interaction, and this kind of exploitation can lead you to something that was not possible until now. That's why we designed our sensor with extreme sensitivity And when I talk about extreme sensitivity here in this work, I'm thinking of, small concentration, ultra low concentration of ultra low molecular weight molecules, and in particular nucleic acids and proteins. And in this platform that you can see here that we call Cancer Radar, we start from the question which therapy draws. This is a platform for liquid biopsy, and the circulating tumor cells are collected, are vested, enriched, analyzed, and what we usually do, we use this technology which is called Destina to, by using bead-based technology, extract these proteins, extract nucleic acids from the liquid, body fluid that can be saliva, serum, blood, urine, whatever, and those proteins are inserted in this microfluidic chip. We have the sensing platform where we have this metamaterial platform for sensing. We measure the shift and look also at the wavefront analysis, and all this goes to a deep neural network, so that at the end we have a bit string that goes to the clinician that characterizes the disease with a list of available molecular biomarkers, and so describe, the list of drug resistances, for example. So how it works, usually what you have is, in a multi-stack, it is an hyperbolic metamaterial, and then on top of it, you have a gridding, which is functionalized with capture molecule. These are surface mobilized so that you have some kind of analytes and target that you want to target. Usual microfluidic channels. We have this all modes excited in this material by shining the material with light, and we measure these ultra deep modes. And what we can see is that as function of, of the material that we inject and is immobilized and is captured at the surface, we measure a shift. We report the shift as function of time, and we have the sensorgram for picomolar, femtomolar, 10 femtomolar, BSA, for example, shows a response in less than few minutes. On 40 minutes, you go to saturation. Well, we measured, when we set extreme sensitivity, we quantified it in 30,000 nanometer per refractive index unit, which is outperforming current sensing technologies. But we know that sensing is not only sensitivity, is specificity, and is also, we have kind of other limits of detection and barriers that we have to break. This is also going all the way down to 10 femtomolar, but the specificity remains our main target now, and that's why we are using chiral metasurfaces for controlling the molecular binding on the surface. What we do on top of these metamaterials, we have, chiral structures, chiral metastructures, creating a metasurface, which are branching out in the fluid, but also they have kind of chirality imbued in this system. And this chirality is such that you can recognize, the chirality of nucleic acids and proteins. To do that, we made some tests. We made some materials, changing the axial pitch, changing the periodicity of this helices, and trying to see if we can gain some specificity, in particular chiral specificity. And so, for example, varying the helix surface coverage, in yellow, you see the surface coverage of biomolecules binding at the surface in different position and with different, surface coverage. And we found that, for example, by doing this test on this hyperbolic metamaterial, a stack of metal and dielectrics, this is our material with the helices, and we, for example, we functionalize this with specific markers with a confocal fluorescence micrograph we analyzed the results, and we found that indeed the molecular binding happening at different levels gives us a different response depending on the coverage. So this has been quantified, and then by using circular dichroism, we compare our simulations, our theoretical effort with our experimental results, giving reasonable good agreement between the results, saying that under superchiral field illumination, you can have some kind of chiral recognition that gives an extra layer on top of biorecognition on top of what we already have as a chemical affinity. In collaboration with Federico Capasso at Harvard, we are working on designing flat optics metasurfaces, which we, in this case, infiltrated with liquid crystal to see how the infiltration process and the forces are enters into play. And for example, here you see the the metastructures and how it looks like in this image. And now, for example, by looking under polarizer, optical polarizer, we use this metasurface and the flat optics to make biorecognition. And in this movie, you see how indeed the infiltration is happening in these flat objects of less than a micron high. So we, by capillary force, we infiltrate this nanosensor, and then we recognize them. These molecules by using the techniques that I showed to you. Well, in the second part, in the next 6 minutes, do I have 6 minutes? Okay.
Yes, about
Okay.
5-6 minutes, it's okay.
Okay, so next, I will try to give you an idea of this part 2, which is another project we are working on at Case Western in the Cleveland Clinic. And this is a weaponizing nanotherapeutic platform for physical killing mechanisms for brain cancer. And you see here, the way in which, during an intracranial procedure for brain cancer treatment, you see the fiber entering the skull and treating, you see here on the screen, this is the skull and the fiber entering in and treating a tumor. And in this case, what we have done is, for glioblastoma, I don't have to mention here what the glioblastoma is, because Gianni Ciofani has told us clearly what's that. It is this terrible monster. Oligodendrocytes and astrocytes and glial cells are in this mosaic tumor cell population, very, very low prognosis and negative prognosis. It's a real monster. So what we have done is try to use some kind of PDT agent, photodynamic therapy agent, based on iridium. This can be excited by light, and during the de-excitation, it excites in presence of oxygen. It excites oxygen, which goes from the molecular state, ground state, which is good oxygen, to the excited state, which is singlet oxygen state, which is terrible reactive oxygen. And so we are using this terrible reactive oxygen, known as also reactive oxygen species, to kill cells when they are in the tumor. So now the idea is, well, take this gold nanoparticles, embed these iridium complexes inside, and deliver to the cells. You see the cells here, which are internalized with these nanoparticles, and this is a platform which you do photothermal, photodynamic process, and imaging in the same platform. And this is just the results of this study, which you see that when there is no illumination and, zero concentration as a control, nothing happens. You illuminate the system, nothing happens. But when you illuminate it, you see the 90% cell viability is less than 10%, which means that 95% of the cells got killed by these physical mechanisms of synergistic thermal and photodynamic processes. But now what we are using are plasmonic nanostructures. You know that a plasmon is a quasi-particle associated to the oscillation of free electrons at the interface between a metal and dielectric. And when it de-excite, it emits photons, so you have light, or can, transfer energy to hot electrons, so you have a non-radiative decay. And these hot electrons increase temperature locally and rapidly. So we are using exactly this effect by designing hyperbolic nanopillar in collaboration with the Italian Institute of Technology, in which we separate the scattering and absorption. We want the absorption to be at a different frequency with respect to the scattering, so that we localize. We know where we have to excite with a different wavelength and be very, very effective in exciting the photodynamic of the photothermal effect separately. And so this is the way in which we did it by using a multi-stack and creating this object. You can recognize here on the bottom left these nanopillars of gold and alumina, and you see that the response is a typical hyperbolic metamaterial response. And what is nice, and these are experiments and theory, the points here are experiments. You see that, for example, the absorption is at 1200 nanometers, whereas the scattering is at 900 nanometers. Saying that, and this is as function of the angle, it's almost independent as function of the incoming angle of the light. So telling that these objects can be internalized, sent there, and independent of irrespective of the angle in which you're illuminating them, they are creating huge absorption or huge scattering of the radiation. This study has been done by using electron energy loss spectroscopy, trying to individuate bright and dark modes in these hyperbolic nanostructures, and by going with an electron beam, looking at its properties, we found that we have several nice modes that can be used for this kind of specific application. And the specific application of this let me say nano antenna and hyperbolic nano antenna is that you can tailor where you want your response, particularly for applications where you try to use light as a way to activate processes in the body, okay? And so to mediate a sort of response in biological method by using light-matter interaction. And this is exactly the way in which I explain these modes. These modes are magnetic and electric modes that are either confined in the medium or localized externally to the structures. And these are, you can see this work in the advanced optical materials where you can look at the details of this. And if you have questions, I will be more than happy to answer your question. Now, I think that my time is over. I want just to tell you that these hyperbolic metamaterials I've proved to possess extraordinary physical properties, can be directly applied to biomedical applications in cancer nanotechnologies. And with that, I thank you very much, and I'm very happy to take any questions that you may have. Thank you.
Thank you, Giuseppe. If there are any questions from the audience, please raise your hands. In the meanwhile, first of all, apologies for misspelling your name before. And okay, if I understand well, your background is a physicist. You are a full professor at the University of Calabria now, and at the same time, a professor at Ohio, a research scholar in surfaces and advanced materials. Is that right?
Yeah, it's correct.
Okay, so you are working with, I have a very general question if there are no questions from the audience. So maybe a kind of an opinion for an expert in the field. having your background in physics and working with metamaterials, you combine different materials to have a specific functionality, right? So shifting this concept to nanobiotechnologies and disciplines, what would you think is the meta discipline to be strengthened in order to address specific issues in nanobiotechnology? So what are the disciplines to put together, and what is the hurdle to be overcome?
Oh, that's a very nice question, but this is the environment in which I work every day. So we go from material science to artificial intelligence to physics, chemistry, biochemistry, and we have also oncologists, virologists. So our team is really molded.
Oh, we lost the right audio. Problems with the audio.
Field in which.
Can you please repeat the last 30 seconds, 15 seconds, because we missed your connection?
Oh, okay, so it seems that my connection is unstable. So yeah, I was telling that we are really a cross-disciplinary field and cross-disciplinary team, and we sit around the same table: material scientists, oncologists, virologists, artificial intelligence people, people working in biochemistry, and many other fields and many other disciplines, knowing that our disciplines are not anymore the disciplines that have been designed in the 19th century, and we work in the ground, and the beautiful fertile ground in between these disciplines, and having a lot of fun there.
So the question is, a specific question is, okay, multidisciplinarity is a key issue, but which link would you like to strengthen between which specific disciplines in this arena?
In my personal opinion, what I like to put together is nanotechnology, artificial intelligence, and nanomedicine, and if you wanted medicine. Okay, and then when you trace a link, a nexus between these three, you realize that you cannot be only these three. You need many more. But this is what I would like to tie.
Okay.
This is the nexus that I'm tying and I'm working in.
Thank you very much. We have time for just another question from Antonio.
Yes. Firstly, thank you very much for very interesting presentation and for having joined this conference. Can you comment on the challenges concerning hyperbolic metamaterials from the point of view of materials? As an example, natural hyperbolic metamaterials or the replacement of silver with low-cost plasmonic metamaterials?
Yeah, that's a very nice question. Thank you, Antonio. I think that the hyperbolic metamaterials are very easy to fabricate once you have the recipe and once you know what you want to do with that. Because the point is what kind of functionality you want to lead, and so that you can mediate an interaction with the biological matter. So now, if you replace one of this material, you enter in a parameter space which is huge, right? And so you want to optimize that. That's why, for example, inverse design by using deep neural network is the way to go. But then you said, well, what about natural materials, natural hyperbolic materials? Well, those are a class of materials which are extremely interesting, but you cannot handle them. Because you are fixed with their properties. We are wavelength range. You cannot tune that unless you dope it. But then this opens a new barrel of worms, because it's doping a material and replacing them is not the same stuff.
Okay. Thank you very much, Giuseppe. So I think we have no time for further questions. There are no further questions. We thank again Giuseppe. Thank you very much. And we can move now to the next speaker of this second batch of the morning session of Advanced Materials for Nanobiotechnology. To introduce Gianni Profeta is a condensed matter physicist. He's a theorist doing first principle calculations, and especially the first principle calculation of the electronic structural properties and also molecular dynamics simulations. So the living matter is composed of 60% of water. This is what we know from textbooks, and so we have to remember anytime that the systems that we are dealing with are composed by water, but it's water confined at the nanoscale, and this is going to be the subject of the presentation of Gianni. So I leave the floor to Gianni. The title of this presentation is Spin Lattice Relaxation Time in Water Graphene Oxide Solutions. Please, the floor is yours.
Thank you. Thank you, Chairman. I hope that you can hear me. And thank you to the organizers for this very kind invitation to this Congress. I start sharing on my presentation, and please give me feedback if there are some problems. So, as the chairman just said, we are interested in water dynamics and NMR relaxation properties when water is nano-confined, where nano-confinement means different kinds of confinement. In this specific talk, we deal with nano-confinement with inorganic materials. This is a joint collaboration with the Department of Life, Health and Environmental Sciences in the University of L'Aquila and with the Department of Physical and Chemical Sciences. And these are the collaborators of this part of the work. So we will speak about, oops, sorry. Okay, we will speak about water. Water is probably one of the most studied systems from different areas in science, in physics, chemistry, biology, medicine, etc. And this is because water presents many different peculiarities with respect to the other simple fluids we have. Here in this graph, I will just report the diverging properties of water as with respect to other liquids. For example, in terms of density, viscosity, compressibility, speed of sounds, and everything, most of these peculiarities are related to the fact that hydrogen bonds in water determine many different features from the liquid to the solid phase. But when water is also put in extreme conditions that can be of extreme pressure or even confined at nanoscale, water starts to exhibit many different novel physical properties that can be also exploited for nanotechnology application, for example, cell biochannels, drug delivery, and water transport in general. One of the main tools to investigate the physical properties and the dynamics of water is NMR, that is the nuclear magnetic resonance, which probes the proton spin of the water and can follow also the dynamics of water in different conditions, even at the nanoscale. And this tool is ubiquitous in science, although we know in medicine the many applications it has, but also in physics, in engineering, catalysis, polymerase science. So it's a very powerful experimental tool to probe the physical properties of water. Here, just present two results that we were interested in the past. The first one is amorphous cellulose that, as you can see, there are many voids that can be filled and emptied by water, and when water is there, the properties of its physical properties changes in many different ways. They can also activate many different reactions and promote different aspects. So NMR can probe the size distribution and also the shape of the voids, as well as the diffusion mechanisms. That is, when water is confined here, in, for example, in two membranes, you can follow the diffusion of water in two-dimensional systems, and you can see that diffusion constant changes as a function of many different parameters. So our aim in general is to study the molecular dynamics of water, modeling the water dynamics at the nanoscale in order to predict the diffusion mechanism and also the relaxation properties of waters in terms of an NMR signal, or in general the relaxation time of the proton spin in an NMR experiment. We did this by means of computational approaches and trying to model the NMR mechanism in when we have the dynamics of water in different contexts. So briefly, I want to show the theory we are using. We use the BPP theory, that is a Bloombergian Purcell Pound theory, which is the state-of-the-art of NMR theory. Just imagine an ensemble of proton spins which are immersed in a constant magnetic field. If we just switch the magnetization of the spins by means of a radio frequency and then wait for the relaxation toward the minimum of the energy, we can follow the magnetization in both the longitudinal and the transversal components of the magnetization, which assume these characteristics shapes, which are characterized by two relaxation times that we call the T1 and T2, which represent the spin lattice relaxation time for the longitudinal component and the spin-spin relaxation time for the transversal component. This relaxation is strongly dependent, and these time constants are strongly dependent on what we call dipole-dipole correlation function. That is, the probability to have two spins at different distance as a function of time. And this quantity, apart the details, is strongly dependent on this P object, that is the probability density to have these pairs of spin exactly at distance r0 at time 0, at a distance r at a later time t. And this probability density determines at the end the dynamics of waters in different constants. And we wanted to access this function in order to evaluate the correlation function and then the relaxation time in NMR experiments. We access this function by means of molecular dynamics simulation, which is a numerical technique in order to probe dynamics in real space. So we solve the Newton equation as a function of time. Once we provide an interaction potential between the nuclei of our systems, and then once we have the complete dynamics of the systems, we can have information about the thermodynamical properties like temperature, like pressure, free energy, total energy, and so on. The force fields developed for water are very sophisticated. There are many force fields which can describe the many physical properties of waters. We choose the T4P potential, which is widely used in the community and is able to reproduce the experimental phase diagram as waters in a very huge in a very large part of the density and of the temperature. So it's quite reliable in predicting many different properties at ambient conditions and even at high pressures at different densities. The potential is quite easy. It just comprises different interaction terms between protons and oxygen, and including also a fixed charge that is used to fit the experimental quantities. Anyway, so once we have the force field, we can now start our simulations and then calculate the trajectories and then use the BPP theory to estimate the relaxation time. So a first example that I want to show is just water. Free water modeled with the 500 of molecules, more or less, and at ambient condition of pressure and temperature. This is the shape of the G star, that is of the correlation function as a function of time in reporting the total correlation time, but also with different colors, the correlation time of intra molecule and intermolecule, the blue fluor, because we have two hydrogens in molecules, so they can correlate at the same time in the same molecule. The diffusion coefficient that we predicted for free water is in very good agreement with the experiments, but what was very important for us is that our implementation gives a relaxation time of 4.3 seconds, which is very close to the experimental measure, the relaxation time T1, that is 4 seconds. So our approach is quite reliable and is able to predict the NMR relaxation properties, which at the end is at the base of any clinical, for example, application of NMR. Once we have now water just well described, we want to ask how we can confine at the nanoscale water. We choose to do that by using 2D materials. 2D materials are a class of materials which are van der Waals bonded, which determine the bonding between different layers, so they can be easily exfoliated, and we can access at different kind of system. Just few are highlighted in these slides, starting from the very first one, graphene, ending with more sophisticated magnetic ones. And in passing, I want to highlight also graphene oxide. It's an allotrope of graphene that we will discuss later more. So the main important properties of these materials determine their use in different fields of science. They can be metals, they can be semi-metals, insulators, semiconductors, they can be strained, doped, they can be functionalized, it can be used for membranes, transistors, coatings, and many other applications. What we think that we try to extend this very long list also for NMR application, and we want to see if they can be used for this scope, or at least can help the physics of the biological physics, including also NMR applications. Indeed, this is not a new field. There are many different approaches trying to use 2D materials for biomedical in general applications, and in particular also for graphene oxide in particular. In fact, graphene or graphene oxide can be functionalized with many ligands or genetic materials or proteins, and can be used for many different applications, as drug deliveries and cancer treatment, and sensing, biosensing, and transport in general. There are few studies also with applying graphene and graphene oxide as a possible contrast agent materials. It means that as a possible material which can alter the T1, that is the relaxation time of water, when water is in close contact with this kind of material. Anyway, these studies are at a very early stage, and we don't know which are the real mechanisms by means of non-magnetic materials like graphene or graphene oxide can induce a contrast agent effect in NMR. So we started to understand how water interact with 2D systems. We just took this as a prototype system and calculate the diffusion coefficients of water when it is confined in these membranes, and when, for example, water is just put on only on a surface of these 2D layers. And from the diffusion coefficients here, is the mean square displacement, we can see that the effect is to slow the dynamics of water, and in a relevant way. And among the different materials we used, graphene oxide is one which is the best in this control. Graphene oxide, as I told you, is the material is derived from graphene with functionalized with different epoxy and hydroxy ligands to graphene. And now we can see what happens when we have one layer of graphene oxide immersed in water in terms of the relaxation properties. We see that nothing changes so much with respect to water. Even if you increase the concentrations, and the relaxation time is practically unchanged with respect to pure water. However, when we have many of different layers, meaning different flakes, we can have the possibility to join two layers in a point line plane mechanism, and then at the end we end up with a bilayer. So we decided to extend the study to the bilayer system, including the solvated system and also as a channel to transport water. And now we found that a relevant difference with respect to the pure water, in particular when we have water just filling the bilayer GO. In this case, the relaxation properties changes a lot. In particular, if we look at the blue curve, it shows the first part as a function of time, which is characteristic of a free diffusion mechanism, but at later time, starting from 100 picoseconds, we have an activation of a Levi flight in which water is strongly interacting with the graphene oxide surface, and the diffusion is strongly hindered. Fourier transforming these curves, we have the power spectrum as a function of the frequency, and from them we can estimate the relaxation time. And in the case of graphene oxide bilayer, we predict a strong reduction of the relaxation time, which now pass from 4 in the case of free water to 1.5 or 5.4 for in the case of water filled with filling GO bilayer. So we predict a strong contrast effect. It means that when water is close to fills the graphene oxide bilayers, its relaxation properties strongly changes. So graphene oxide can have these contrasting effects, and the contrasting effects is well explained by what we call two model systems, two fluid system, when we have two kinds of waters, one interacting with the surface and one with the bulks, exchanging during the dynamics. So I think that I have to conclude. So the conclusion are that we have discovered contrasting effects in 2D materials when water fills the bilayer systems on these systems. Only the confinement geometry is able to shorten the relaxation time. Now we can extend the study in order to include magnetic systems, that is just doping the graphene or graphene oxide with magnetic or paramagnetic atoms, can then exploit this confining mechanism, also including the possibility of magnetic interaction, for example, also using magnetic 2D systems that were recently discovered. So I hope I'm in time, so I thank you for your attention, and I open for questions.
Thank you, Gianni. Time for just one quick question from the audience. Yeah, there's one from Mariangela Di Donato, please.
Hello, thank you for your presentation. Can you hear me?
Yes.
Okay. So thank you for your presentation, it was very nice. Just a short question. Which is the distance among the two layers of graphene oxide? So,
okay, good question. Sorry.
No, I was because in your last slide you were showing that you have bulk water and confined water. So you have both that kind of water in the layer.
Yeah, okay, I missed the information. We use 1.5 nanometer distance between the two layers. And we also have studied as a function of the distance. And what is very interesting is as a function of the distance, water can have also a super diffusion mechanism, in which when the distance is reduced too much, the diffusion coefficient increases with respect to the free water. This is very interesting properties that we are now trying to exploit in different contexts.
And do you think, just related to that, do you think that this is because the connectivity of water changes? Because
Yeah, exactly.
Okay. Thank you very much.
Thank you. Are there any more questions? If not, I have a question myself, and it is related to, do you have any supporting experimental evidences of your MD study?
Yeah, there are some experimental evidences already present in the literature, but there are studies in which the contamination of graphene oxide in some sense hinders the nature of contrasting effects of pure graphene oxide. We here in L'Aquila tried the same experiments, including different purification of graphene oxide in order to see the real effect of this confinement effect. And indeed, we found some supporting experimental information along this line, in which just graphene oxide is able to have a relevant contrasting effects. They're not in compared with gadolinium chelates, it's sure, because we don't have magnetic systems. But now we have the possibility to have a very large system in which can be also functionalized and can be used for theranostic. That is, you can see your damaged part, let's say, of a body, and then trigger, see with the NMR, and then release the drugs exactly where they are. So it's, I think that it could be used for that applications.
Yeah. Thank you very much, Gianni. So there's no time for further questions, so we quickly move to the next talk of the day, which is a pre-registered one from Ilaria Tonazzini. That's from CNR Nano and Scuola Normale Superiore di Pisa. And the title of the presentation is Directional and Asymmetric Microstructured Substrate for Applications in Neuropathological Models. Okay.
Good morning. My name is Ilaria Tonazzini. I'm going to present you our work about directional and asymmetric microstructure substrate for application in neuropathological models, in particular for regenerative application. Nerves constitute a very rich web in our body. Nerve injuries are a common trauma with high incidence and impact on healthcare system, affecting more than 1 million people every year. Their regeneration is a complex phenomenon that today still presents several research and clinical issues. In fact, besides the advances in the repair methods, such as the use of artificial conduits, the use of autologous nerve graft with the relative site donor morbidity is still the gold standard in clinic. And after this, the functional recovery usually is far from being satisfactory. Artificial scaffold can offer new opportunity for nerve regeneration application, and in this framework we can play with the topography and the chemistry of the material and the scaffold. Here, controlling and improving neuronal and Schwann cells migration and growth are critical elements for nerve application. And here, Schwann cell, the glial cell that unsheath the axons of nerves, have a crucial role And in fact, Schwann cell migration from the proximal stump is one of the first physiological mechanisms promoting axonal regrowth after nerve injury. In our work, we focused on the study of cell-material interaction and in particular on the topography. By different microfabrication technique, we engineered nano and microstructure membranes in thermoplastic or elastomeric material with a pattern of grating, so alternating lines of ridges and grooves of varying dimensionality, so varying width and depth, with the aim to induce directional stimuli to cells. In previous work, we showed that the interaction of grating with neuronal cells promote bipolarity and neurite alignment to the substrate topography by the geometrical constraint of focal adhesion and their crosstalk with the cell actin contractility. Gratings have been demonstrated to be effective in directing and guide several neuronal cells according to their period size, both at single and network level. We then exploit grating with different periodicity to tune Schwann cell guidance with the aim to find the best topography to enhance nerve regeneration. Schwann cell responded to grating topography with the alignment and the cytoskeletal shaping in accordance to the pattern and directional migration, but in particular substrate with the T4 periodicity, so 2 micron groove and 2 micron ridges resulted the most effective pattern to guide and promote Schwann cell invasion. Regarding the scaffold material, regenerative medicine is searching today for new biocompatible, possibly natural and biodegradable polymer materials. Here, chitosan is emerging as a promising biopolymer thanks to its characteristic, and importantly, it is also FDA approved for several medical uses. We recently showed that it's possible to obtain by solvent casting chitosan thin film with good mechanical properties and to structure them at the microscale and down to the nanoscale. So we obtain a grating with a period of 200 nanometer. Today, I'm going to show a simple and effective method for the production of microstructured chitosan film by solvent casting in 2 steps with 2 molds. This method has high fidelity, micropattern reproducibility, and is performed in aqueous solution without solvent and completely at room temperature. Thanks to this method, we developed 3 types of chitosan microstructure with grooves patterns with different level of symmetry. Classical gratings with 2 symmetry axes, a zigzag pattern composed by isosceles triangles with 1 axis of symmetry, and zigzag pattern with scalene triangles with no axis of symmetry. And the period was 10 microns, so 4 micron ridge and 6 micron grooves. The topography were chitosan membranes were then characterized using atomic form microscope and also after exposure to liquid environment and to cell culture condition. And we found that our chitosan film maintained their structure and the topographical microfeature were unaltered after exposure to liquid and cell culture for several days. We then tested our membranes with the Schwann cells, in particular with the Schwannoma cell line RT4 cells. As you can see, cells grow optimally on our substrate, reaching also confluence. And in particular, we find an increase in cell density on the SCA topography after 24 hours. We performed immunostaining and confocal microscopy to study how topographies influence cell morphological feature parameters. We looked at actin fiber organization by phalloid staining, at the cell-cell junction by encadening staining, and nuclear morphology. You can see here how all the topography are effective in inducing cell polarization, aligning both actin fibers and cell-cell junction along the pattern direction. But here, SCA polarizes and guides less strictly the Schwann cells in front of the other topography. Regarding the nuclear morphology and nuclear alignment to the pattern, again, SCA leaves a higher degree of freedom to nuclear orientation in front of the other substrate. Individual cells were traced and measured for cell elongation and alignment angle to the pattern. Cell alignment is here reported as the average angle between the cell main axis and the pattern direction. This means that good alignment corresponds to a small angle, while random outgrow, like on flat, gives high alignment angle around 45 degrees. Schwann cells are guided by the underlying directional microtopography, but also shaped according to their asymmetry. In fact, again, SCA leaves the highest freedom to the cell shaping in respect to the other topography, with values that are intermediate between flat and gratings. We cultured cells on our Kitson microstructure films, and we measured single cell migration for 24 hours by time-lapse live microscopy. The Schwann cell steps aligned to the pattern, so with an angle between 0 and 15 degrees, increase in all microstructured membranes. Even if all induce a directional migration, only SCA is able to enhance the final displacement reached from cells. Moreover, Schwann cells on SCA persist for more time on one specific direction and are less likely to reverse their migration direction. So overall, SCA improves Schwann cell migration at single cells and promotes further displacement of the cell and an increased persistence in one direction. We look at then collective cell migration with dealing, and we followed for 20 hours on the pattern. Again, SCA is the best performing pattern and improve collective migration in front of standard flat surfaces. So, SCA leaves the higher freedom to cell shaping, but improves most cell migration. Why? To respond to this question, we performed an analysis on the terminal protrusion of Schwann cell on our microstructured surfaces by measuring the ratio between the two cell protrusion areas. The two that you can see in these graphs. The two main cytoplasmatic edges extend equal on grating in both sides of the cell, resulting in a similar extension of both ends. And this happened also on its substrate. The symmetry of the two cytoplasmatic cell ends is instead limited in cell growing, developing on SCA substrate. And here one cell is always larger, one end of cell is always larger than the other one. And this asymmetry leads to an increase in the area ratio between the two protrusion edges. So SCA induced the establishment of an asymmetric cell front profile in Schwann cells. We also analyzed the actin fiber intensity signal in the two cell ends of each cells has an index of actin protrusion contractility asymmetry index. We found a rise on SCA of this protrusion asymmetry index. And overall, We think that SCA inducing an asymmetric profile in cells induce also an asymmetric organization of actin in the protrusion of the cells. This likely caused the higher cell migration and directional persistence of cells on SCA. Concluding, I hope I showed that we obtain chitosome film with the macrostructured patterns by solvent casting with a simple, reproducible, and cost-effective method. The less symmetric topography SCA polarize less efficiently cell shaping, but promote a faster cell migration of cells both at the single and collective level. So overall, SCA is the best performing substrate for healing of nerves. And our result overall allowed the identification of specific topographical elements that may be exploited for the production of new scaffold for improving peripheral nerve regeneration. In the end, I would like to thank my group in Pisa, Neuroscience, and Luca, Roberta, and Alessia, that are the person mainly involved in this project, our collaborator in Turin, and you all for the attention.
Thank you, Ilaria. And if I understand well, Ilaria, you're online, right? So
yeah.
It has been pre-registered.
Yeah, they said it was the preferred way, so I did just for that.
Nice. So the time is okay. The timing is okay. So if there are any questions from the audience, no. Okay, but I have a question. So I have the privilege, being chairman, to be curious. And as I understand well, you studied the directed growth of cells by substrates which have an asymmetry as a substrate. Okay, which is very much interesting, by the way. The question is, did you try, as you've studied at Rotterdam in the Neuroscience Institute, if I understand well, did you try with stem cells? Because something has been carried out here in dell'Aquila of the differentiation of stem cells on graphene oxide, but this was not a substrate which had a broken symmetry, a directional broken symmetry. Is there any study in this sense?
Yeah, we did, because another line, let's say, of research in the group is to use this nanostructured and directional material to test the topographical reading of primary neurons. So we studied this, the response of neuronal cells, both in human inducible neurons, so neurons that have been developed from stem cells from patients and human controls, LTE persons, let's say, and we did also with primary neurons from mice from different type of neurodevelopmental models. And we didn't test the different asymmetric pattern, but just mainly the gratings. And we see that when there are some defects, we can have some defects at the level of connectivity in the pathology. We have some defect in the connectivity and in the guidance of the neuronal cells in our gratings.
Okay, very interesting. Okay, thank you very much. I have no further questions.
Okay.
There are raised hands from the audience, which I see Mariangela Di Donato, who has switched the screen. Mariangela, do you have a question?
No, it's just because I have to talk after that.
Okay, bye, thanks.
I will just have to.
Thanks,
Okay.
bye.
So we thank again Ilaria, and it was a very interesting talk. And so we move to Mariangela. I will introduce you later, so I give you the floor immediately. Mariangela Di Donato from CNR Ecom Sesto Fiorentino, and the title of the presentation is Addressing the Photo-Induced Mechanism of Visible Operated Molecular Photo Switches. The floor is yours.
You can see my screen?
Yes.
And try to start the presentation. If it works, doesn't look like. Okay. So, okay, first of all, I want to thank the organizer for giving me the opportunity of talking about our work on visible operated molecular photoswitches and understanding their mechanism. So, photoswitches are molecules which can respond to light by changing one of their photochemical properties. Like most of the cases, they change their structures. So, they can undergo, for instance, to assist trans isomerization. And since these molecules can be sensitive to light, they can be embedded in a material or a nanomaterial and confer them some optoelectronic or optomechanic properties. Photoswitches can be used in numerous applications. Here I just show a few of them. For instance, they can be used to fabricate molecular machines. They can be embedded in polymers and give them some motion properties. They can be used in photoactivated catalysis. They can be used for constructing optical memories, or they can be used also in biomedical applications, such as photopharmacology, where we can activate a drug precisely in its site of action by light. And most of the photoswitches that are currently used are activated by UV light. Think, for instance, azobenzene, that is one of the most famous and most applied. But for several applications, in particular for the ones that are dealing with nanomedicine, it will be very useful to have photoswitches that are operated by visible light. And here I will give you just a couple of examples of molecules that we recently studied to understand the mechanisms that are activated by light. So the first molecule that we studied is this donor acceptor phenase adduct that is shown here. This is a modular molecule which is formed by a donor part and acceptor part, and they are connected by a tri-end chain. And this molecule absorbs invisible, and upon light absorption, they undergo a large structural change and form this cyclized form that is instead not colored and absorbing the UV. And one of the nice things about these molecules is that you can change the donor and the acceptor part and shift very much the absorption spectrum, going up to the infrared region. And so we were interested in understanding the mechanism by which this photoreaction occurs, so how to pass from this open form A to the closed form B. And the mechanistic hypothesis was that the molecule, once light activated, could undergo isomerization first and then ring closure. So our question was, can we observe some intermediate in this reaction and with some spectroscopic tool? So what we started from was the observation that if you irradiate the molecule and measure the absorption spectrum, you will notice that while the main absorption band decreases, a new band increases on its right side. So this could be a band of an intermediate, but we have no further information about that. So we wanted to characterize the time scale of appearance of this intermediate and this nature. So we did some transient absorption spectroscopy measurement, irradiating the sample with the laser 520 nanometers, and we noticed that on a very short time scale, while the main band was decreasing, a new band of photoproduct was appearing on its right side. So by analyzing this data, one can extract the time scale of appearance of this product band, and here you can see that it's very fast, so the product is appearing in about a few picoseconds. But we have no structural information about these photoproducts from this measurement. So we performed some infrared measurements, also time resolved. And here you can see the spectra. So these negative bands well correspond to the FTIR spectrum of the molecule. And after some time, you see that there are some product band rising. And these are the bands of the intermediate that we want to characterize. Once again, the analysis gives us the time scale that, of course, are very similar to what we found before with visible spectroscopy. And now we look at the long-living component, the one that is telling us which is the structure of the photoproduct. We see that the main band shifts are in these two regions, so 1100, 1200, and 1500 numbers regions. And these regions are characteristic for cis-trans isomerization. So at this point, we have a more clear idea about the mechanism, but still not complete picture, because to go from this open structure to this closed one, the system has to pass through this intermediate a second. And there are two ways to go to this intermediate. One is a direct Hula twist isomerization, in which we rotate both a double and a single bond. And the other mechanism is a sequential first double bond isomerization, then single bond rotation. In order to discriminate among these two mechanisms, we did some calculation. Actually, our collaborators did it. So they computed the structure of the three forms that we have here, so the initial one and these two intermediate. They computed the infrared spectra, and we compared the experimental and calculated results. And from this comparison, it is clear that the mechanism is sequential, so because the agreement is much more relevant with the structure of A prime, so this intermediate here. So from this, we conclude that the mechanism proceeds to a sequential scheme. We first have double bond isomerization in about tens of picoseconds, and then on a longer time scale, we have single bond rotation and ring closure. Actually, this we also studied this step of the reaction that is much more complicated than what I'm showing here, but for time reason, I will not show you these results. If you're interested, you can look at this publication. The second molecule I want to talk to you about is this new photo switch that was recently synthesized called ITI. And this was synthesized with the aim of having a switch that could be applied for more medicine-oriented applications. So we aim at something that could be used for photopharmacology, and the properties that a switch should therefore be used in this field is that it should be fully activated by visible light, so the reaction could proceed in both direction with the visible light. There should be a large separation between the bands of the two photoisomers in a way to address the reaction specifically from one side and the other. We should have control of the isomerization time, and the molecule should be easy to synthesize. So this molecule that we are going to study is depicted here and is basically formed by alpha-hydrobenzene alpha-thioindigo. And it undergoes a ZE isomerization upon light absorption and absorbing the visible spectral region. We did some first ultrafast characterization to understand the time of this isomerization reaction, and we find out that isomerization, photoisomerization is ultrafast. So the mechanism is very similar to what you find for azobenzene, and the reaction proceeds on a really ultra-fast time scale, less than 1 picosecond. We have the formation of the isomer. As you can see here, we see a product band that is redshifted compared to what is the absorption of the initial isomer, and the band separation is very large. So this is a nice property because now we can switch selectively the two conformations. So we did some calculation as well to understand why there is this large band separation. And the results of the calculation tell us that the E isomer has a banded structure, and this is destabilizing the HOMO orbital. So this is because of this destabilization, the band gap between the HOMO and LUMO decreases, and so the absorption of the E isomer is redshifted. We also measure the time necessary for the reaction to go back, so from E to Z. And we see that the time scale is in the millisecond time scale at room temperature. So the reaction is very fast, also the back reaction, which limit a bit the application of this system. And but going at low temperature, it is possible to photoaccumulate the E isomer, and you can see that there is a change of color in this solution. And so at low temperature, the system can also be back switched by light. But we wanted to go further and see if it's possible somehow to increase the lifetime of the E isomer. And one of the ideas that we had was to engage this nitrogen with a covalent bond. And we started to see if by protonating this nitrogen, we could increase the lifetime of the E isomer. So the first observation we had is that by protonating this nitrogen, so adding a strong acid to a solution, we see that the main absorption band of the photo switch red shifts, and this is already nice because we go even more in the red. In particular for this species in which we have a methoxy substituent on the benzene ring, we have a very redshifted band. And we first studied the photochemical part, so the photo switching mechanism. For the normal ITI, things are very similar for protonated and unprotonated species. So once again, we have a very fast switching on a sub-picosecond time scale. For this substituted molecule, the spectra changes a lot, and also the kinetics is a bit slower. But what is important is that already by adding 230 millimolar of acid, we can increase the lifetime of the E form to 15 milliseconds. Then we also did some further calculation to understand the mechanism by which this photoreaction is occurring. And calculation told us that when you excite the protonated molecule, isomerization occurs in the excited state. Then what happens is that the system reaches a conical intersection, goes back to the ground state. And here, back reaction is not occurring directly from the protonated species because there is a very high barrier to overcome. So the system first has to deprotonate and then it can go back to the Z form. So this mechanism tells us that by changing the pH of the solution, we could also change the lifetime of this isomer. And this is indeed experimentally proven because by adding an increasing amount of acid, we see that the lifetime of the isomer increases by several orders of magnitude, reaching seconds starting from milliseconds. And here we also show that both isomers can be interconverted by light, so we can convert we can do both cycles with light using visible light in both directions. So with this, I will briefly conclude my presentation just with the short message. And the message is that when we look for a switch and want to improve the properties, it is very important to understand the photochemical mechanism because this tell us which changes we could then make to the molecule in order to improve their properties. And then we also show that a multidisciplinary approach based on chemistry and chemical spectroscopy and theoretical calculation can really be informative about the mechanism of this photochemical reaction. And with this, I want to thank my collaborator in LENS and ICOM, and my external collaborator, especially the group of Professor Benferinga and Professor Witten Szymanski, or the Professor Bibernian Boom as well, and the calculation part that has been done by Professor Claudio Filippi, Dr. Adelovan, and Dr. Miroslav Medved. And thank you for your attention, and thank you to Lateral Europe for funding. I can't hear you.
You're right. I had my microphone switched off.
Yeah.
So thank you very much for staying in time, and there's room for a couple of questions and then we're over. But in the meanwhile, if I would like to stimulate the audience. But by the way, your background is in chemistry, if I understand well. And so you started from the University of Salerno, then you moved to Sesto Fiorentina and then to Amsterdam. So you're a nice example of cross-contamination of disciplines, and because I was looking at your CV and then I wondered how come she's a chemist and how come she's doing optical properties, and then I understood a little bit from your presentation. So the question is, as this is a session devoted to nanobio applications, I don't know if I missed something, but would you stress a little bit the possible potential application of your studies in nanobio applications?
Yeah. Actually, I only presented, let's say, a very basic photochemical study about these molecules, but the first example I showed, the DASAs, have been applied in several systems. For instance, you can use this molecule in vesicles or nanoparticles, and since they undergo this large structural change, when you illuminate, you can basically destroy the nanoparticle, the vesicle, for instance. So you can use this system to transport, for instance, a drug or also a molecule from one solution in which it is soluble, from one phase in which this molecule is insoluble to a phase in which the molecule is soluble. So as a transport media, they have been used. You can also embed these systems in polymers, for instance, to confer them some property like motion properties. And the second example I've shown is really new molecule, and the aim is to apply it in some more biomedical-oriented application, for instance, incorporating this kind of switching protein. At the moment, this is still like a dream because the molecule has some properties that have to be optimized, but the idea is to incorporate it in either in proteins or, for instance, attach it to a drug and confer it some light sensitivity.
Yeah, very interesting. Thank you very much. I don't see any further question. So I thank you, Mariangela, again, and I take the chance to thank all the speakers of this morning session, which has been very interesting. And let me switch off the phone. And there was an average of about 30 participants and attendees to this room, which has been quite crowded, even being a virtual room. So I thank the audience for being so careful and attentive, and I would like to thank once again the organizer for giving us the opportunity for exchanging ideas at the state of the art and cutting edge research in nanobiotechnology. I'm quite surprised of the quality of this session, and I would like finally to thank also the technical organizers for this perfect organization of the event. I don't know if there is a word from the organizers. If not, I'd like to thank everybody, and this session is then closed, and have a good lunch, everybody. Bye-bye.
Good afternoon, ladies and gentlemen. It's a big pleasure to introduce this section. The title is Advanced Material for Water Treatment and Environmental Sustainability. It is also a big pleasure for me, also because for my research activity, this topic is very related to my work. I am Francesco Deglio. I am full professor in chemical engineering at University of L'Aquila, and my main research activity is related to hydrometallurgical process to recovery precious metal, rare earth, base metals from e-waste or industrial waste, but using hydrometallurgical operations. So we use water with chemicals. And in the development of our process, very often we use also wastewater treatment section in order to reuse water, for example. It's a very important topic, even because also my second activity is mainly related to industrial wastewater, especially industrial wastewater. Just to give you some idea about the importance of this section. For example, now actually we are working with food industry in which we need to resolve the problem of the presence of sodium chloride in the wastewater and also polyphenol using an integrated approach with membrane process and biological process or advanced oxidation process. For example, is another company, galvanic company, that also is interested also in the water reuse. After the galvanic process, they need to remove heavy metals, and also the presence of chloride is very difficult to remove. The only way is to use an integrated approach with membrane, for example, evaporators. Another example, also, we worked in the past in the ambit of a LIFE project with a microelectronic industry, in which they had the problem to dispose some waste in the presence of water, with organic compound. We have developed a biological process integrated with a membrane process in order to optimize the design of bioreactor. But also the example, just 2 days ago, I had some contact with a company that is interested to the treatment of water that is produced by oil extraction, in which they need to remove boron. boron is also a critical element for European Union. So to change the idea to avoid waste disposal, but to implement wastewater treatment. Other example, I think I don't want to spend a lot of time, but finally, last but not least, our process in which we develop process, we realize pilot plant, industrial plant to recovery precious metal rare earth from some kind of waste, for example, printed circuit board, spent batteries, spent catalyst, etc., using hydrometallurgical operations. So at the end of the process, it's important also to develop suitable and sustainable technology in order to improve the circular economy of the process that we have developed. So this is just a very short introduction about this section, and also to present myself. And I think now we can start with a short presentation of our minister, Roberto Cingolani, is our Minister of Ecological Transition. So that he will present some ideas, some words for us before to start our keynote section. I ask to Jessica if it's possible to start with the video. Thanks.
Cari ex colleghi, salve a tutti. Mi fa piacere salutarvi per questa First International Virtual Conference su Material Science and Engineering. Non mi aspettavo in questo punto della mia esistenza di cambiare così repentinamente mestiere, sia pure per un periodo molto breve, però devo dirvi che pur trattandosi di una attività da ministro, comunque continuo ad avere a che fare con tutte le cose che ho fatto e studiato, peraltro con molti di voi, durante questi decenni. Probabilmente la grande chiamata che ci viene fatta dagli organismi internazionali per la transizione ecologica sarà uno stress test di proporzioni epiche per tutto il comparto ricerca, sviluppo ed educazione. Sull'educazione premetto che dovrà partire una campagna che durerà almeno 2 decenni per conseguire la cosiddetta public awareness e dovrà partire dai giovani, dai giovanissimi, dalle scuole. Non esiste, credo, la possibilità di avere una transizione ecologica di successo se non creeremo delle nuove generazioni che capiscano profondamente quello che sta succedendo alla termodinamica del pianeta, all'acqua, alla terra, la chimica di quello che mangiamo, di quello che coltiviamo, e queste cose purtroppo non si può aspettare il 20° anno di età per studiare all'università, bisogna cominciare a spiegarle in maniera semplice a 6, 7 anni, 8 anni, anche approfittando delle tecnologie digitali. Su questo ognuno di noi come genitori in primis e poi come insegnante, ognuno di voi, io purtroppo non insegno più, ma insomma può aiutare a far crescere la consapevolezza contro l'ideologia e i movimenti di chi molto facilmente ritiene di avere soluzioni immediate per problemi lunghissimi. Dal punto di vista tecnico-scientifico, quello che ho potuto constatare in questi mesi è che i problemi con cui abbiamo a che fare sono salvaguardia delle riserve idriche, dei mari, degli ecosistemi, utilizzo intelligente delle terre, rischio idrogeologico, chimica dell'atmosfera, politiche energetiche, vettori di energia, sistemi di storage, come trasformare l'energia. Ovviamente chimica dell'atmosfera è anche molto importante dal punto di vista dell'azione antropica, dell'utilizzo dell'energia. Tutta la chimica dei materiali, che viene chiamata con un termine forbito economia circolare, ma in realtà è la chimica del materiale, la nostra Material Science and Material Chemistry. Qui ho capito che molte delle cose che io stesso pochi anni fa con alcuni di voi sviluppavo, le spugne nanotech per separare l'acqua dall'olio, certi sensori ad altissima sensibilità che erano ibridi fra biologico e inorganico, le chimiche biodegradabili per le plastiche a base di vegetali, di carta, certi sistemi di storage basati su batterie o fuel cells che utilizzavano contatti micro e nanostrutturati, chimica degli elettroliti, tutta questa roba qui che sembrava essere un divertimento di laboratorio, in realtà adesso che sono dall'altra parte della barricata sta diventando sempre più importante. Non ci sarà transizione ecologica se non ci sarà sviluppo della ricerca e dell'innovazione. Credo che questo sia un compito che dico ancora alla nostra comunità, perché spero di terminare presto questo ruolo e tornare a fare il mio mestiere, che la nostra comunità dovrà in qualche modo servire e assolvere in una maniera molto costante, dedicata, mai come in questo periodo, direi da secoli a questa parte, il ruolo degli scienziati e degli sviluppatori assume un'importanza fondamentale per il futuro, non delle prossime generazioni, ma della generazione di bambini che abbiamo adesso nelle scuole. Quindi vi auguro buon lavoro, divertitevi voi che potete, perché anche io mi divertirei molto volentieri a partecipare a una conferenza in questo momento. Spero di poter vedere alcune delle vostre cose subito applicate ai tanti problemi che arrivano su questo tavolo in questi giorni, dall'immondizia all'acqua, alla depurazione, alla chimica dell'atmosfera, come dicevo prima. Quindi conto molto su di voi e sul vostro aiuto. Buon lavoro a tutti.
Okay, many thanks to our minister for a very nice word. I think all the topics that are interested for this ecological transition have been introduced by our minister. So I think that it's time to start with our keynote speaker. And so we start, okay, we have in time. It's a pleasure for me to present Giuliana Impellizzeri. She has a permanent position as a researcher in the CNR, IMM, Institute of Microelectronics and Microsystems, since 2004. She obtained the master degree in physics with laude from University of Catania in 2000. She worked as a device engineer at ST Microelectronics for 20 months. She attained the PhD in physics at University of Catania in 2004, and she is author of more than 150 papers in international reference journals. So the topics of this keynote is a novel photocatalytic nanocomposite for water remediation. So another important subject in the wastewater treatment. Please, Dr. Impellizzeri.
Good afternoon. Before starting, I would like to thank the organizers for this kind invitation. During this speech, I'm going to show you some of our recent results based on photocatalytic nanocomposites for applications in water remediation. The second top problem for humanity is water, and the World Health Organization estimated that by 2025, half of the world population will live in areas of high water stress. In addition, in view of the circular economy principles, it's important a sustainable water management. Thus, it's crucial to find new strategies to recycle wastewater efficiently in a low-cost and eco-friendly manner. Heterogeneous photocatalysis is one of the emergent and innovative methods for wastewater treatment. When a semiconductor photocatalyst immersed in polluted water is irradiated by photons with energy equal to or higher than its band gap energy, the produced electron-hole pairs can cause the generation of reactive oxygen species on the photocatalyst surface. The reactive species promote redox process, which destroy organic compounds and damage microorganisms possibly present in water. Photocatalysis is so promising because it is effective in degrading refractory organic compounds and water pathogens. It operates at ambient temperature and pressure. It can induce a complete mineralization of contaminants without the production of secondary pollutions. It has low operating cost. The photocatalyst can be reused countless times. Nowadays, one of the major technological obstacles hindering the commercialization of photocatalysts is the recovery of the photocatalytic powders after the wastewater treatment. Photocatalysts in the form of nanopowders are highly efficient due to their high surface areas. However, nanoparticles can be extremely toxic and the separation process is required after the treatment. The incorporation of photocatalytic nanomaterials in a polymeric matrix is a promising strategy to solve this problem. Polymeric nanocomposites have recently attracted great attention since the presence of the nanomaterials, the immobilization of the photocatalyst, the low cost, and the flexibility of the polymer. This could open the way to new applications. Zinc oxide was the catalyst of our choice because it has high photocatalytic performance, it is environmental friendly, it has a low cost, it's easy to synthesize, and it has band position favorable to redox reactions. The polymers of choice were polyethylene naphthalate and polymethyl methacrylate. They are used in many common applications for their transparency to visible light, their chemical stability, good mechanical properties, and low cost. The photocatalytic activity of the investigated materials was evaluated through the discoloration of methylene blue dye under UV light irradiation. The samples, 1 cm² in size, were immersed in a solution containing methylene blue and ionized water. The mixture was then irradiated by a UV lamp. The solution was measured at regular time intervals with a spectrophotometer. The concentration of methylene blue was evaluated by the absorbance of the methylene blue peak at 664 nanometer using the Lambert-Beer law. Before starting with the UV irradiation, the physical absorption saturation level in the dark was reached for each sample, so to discern between the absorption phenomena and the photocatalytic ones. This preconditioning step was performed for all the photocatalytic tests reported in this presentation. We also tested the degradation of other water pollutants such as phenol, drugs, and personal care products. The nanocomposites, objects of this presentation, were realized thanks to the atomic layer deposition. It's a vapor-based deposition technique in which highly reactive precursors are pulsed alternatively into a reactor chamber where a substrate is placed. Each pulse is separated by a purging step with an inert gas. This technique relies on the self-limiting reactions between the atomic layer deposition precursors and the reactive groups on the substrate surface. The atomic layer deposition offers an excellent uniformity, excellent conformality, high reproducibility, easy scalability, and low deposition temperature to obtain polycrystalline materials. This last property is important due to the thermally fragile nature of the polymer. Atomic layer deposition on polymer is a challenging process due to the absence of hydroxyl groups on their surface that are in principle necessary to start the growth. In this work, the group of Wilson succeeded in depositing aluminium oxide on several polymers, and they also proposed a simple model to predict the deposition. 3-methylaluminium and water were used as precursor. The idea is that the nucleation of aluminium oxide is promoted by the 3-methylaluminium diffusion into the porous structure of the polymer and the subsequent reaction of the retained 3-methylaluminium with the water molecules. Carrying on the deposition, an aluminium oxide film grows on top of the polymer surface. Here I reported the main chapters of the talk. We firstly performed a systematic study of zinc oxide films deposited by low-temperature atomic layer deposition on silicon. Then the process was transferred to polymer substrate. The coating of PMMA powder by zinc oxide realized by atomic layer deposition was exploited. And finally, the coupling between silver nanoparticles and zinc oxide films was studied, so to boost the photocatalytic performance of zinc oxide. The first study was dedicated to the deposition of zinc oxide by atomic layer deposition at low temperature on silicon substrates. This slide reports the diffraction patterns of zinc oxide films with different thickness deposited at 80 degrees C. All the zinc oxide films were polycrystalline despite the low temperature, and in particular the XRD confirmed the Wurtzit crystal structure of the samples. This figure shows the degradation of methylene blue dye as a function of the radiation time. The concentration of methylene blue at time t is denoted as C, and at time 0 as C0. We tested the discoloration of methylene blue in the absence of any photocatalytic materials and in the presence of zinc oxide films with different thickness. No response was obtained without any sample as expected. Instead, the zinc oxide films induced a significant degradation of methylene blue. The increase of the photoactivity with the film thickness is a consequence of more charge carriers generated by photoabsorption. It's worth noting that the photoactivity tends to saturate for high thickness. According to the Langmuir-Ishwood model, the photodegradation reaction rate of water contaminants is given by the present equation. This plot reports the reaction rate as a function of the film thickness for zinc oxide deposited at 80 degrees C. The maximum activity was reached at about 40 nanometers. This means that for thick films, the charge carriers generated deeper than 40 nanometers from the surface do not reach the surface. I mean, they recombine before reaching the surface, and as a consequence, they do not contribute to the photoreactions. In order to quantify the charge diffusion length normal to the surface, we fitted the increase in photocatalytic activity with increasing film thickness by an exponential law. K is the measured ray constant, lowercase d is the film thickness. This investigation demonstrated the importance of the film thickness and should be taken into account for the production of more efficient zinc oxide-based photocatalyst. Afterwards, we transferred the atomic layer deposition of zinc oxide on polymers. After a careful optimization process, we obtained a zinc oxide film deposited at 80 degrees C, 50 nanometer in thickness on PN substrate, as reported in this CM image. The zinc oxide film follows the morphology of the polymer, as expected since the conformality of the atomic layer deposition process. The zinc oxide film showed a significant reduction of methylene blue irrespective of the substrate, PN or silicon. To demonstrate that the synthesized material is also active in the degradation of dangerous organic pollutants, the performance of the materials was evaluated through phenols. Phenol is a highly toxic compound used in many industrial processes. Here, the percentage of the phenol degradation after 4 hours of UV light irradiation is reported. The films of zinc oxide deposited on PN or on silicon were able to remove the 30% of phenols present in the solution. In this experiment, we coated the commercial PMMA powders with a thin layer of zinc oxide. The thickness of the zinc oxide coating was estimated by transmission electron microscopy at about 80 nanometers. After the deposition, the composite films were prepared by solution casting. Here we can see a photo of a pure PMMA film and a zinc oxide PMMA film. This same image in plan view shows the typical granular structure of zinc oxide. This slide reports the variation in the methylene blue concentration as a function of the radiation time for the zinc oxide PMMA composite films compared to the activity of a mere PMMA film and of a zinc oxide film deposited on silicon by atomic layer deposition, with the same thickness of zinc oxide deposited on PMMA, that is 80 nanometers. The discoloration of pure methylene blue is also reported as control. As expected, no activity was observed without the presence of zinc oxide. The zinc oxide film on silicon was able to degrade the 30% of methylene blue. However, the best results were obtained with the zinc oxide PMMA composite that was able to degrade about the 45% of methylene blue due to the nanostructuration of zinc oxide. In addition, the photocatalytic efficiency remained unchanged up to 7 photocatalytic runs. The synthesized nanocomposites were also able to remove the 30% of phenol after 4 hours of UV light irradiation. A step further towards a superior photocatalytic performance is represented by the enrichment of zinc oxide PMMA nanocomposites with silver nanoparticles. Indeed, besides the antibacterial properties of silver, the combination of semiconductor photocatalysts with electroscavengers such as noble metals increases the separation between the photoexcited electrons and holes, thus reducing their recombination rate. That is one of the main shortcomings of the semiconductor photocatalyst. In this work, after the atomic layer deposition of zinc oxide on PMMA flat substrate, silver nanoparticles were added on zinc oxide by plasma-enhanced atomic layer deposition. The silver nanoparticles deposited on zinc oxide were carefully studied by scanning transmission electron microscopy. This figure shows a plan view TEM image of the sample in which we can see the assembled nanoparticles on the zinc oxide surface. A detailed chemical analysis was performed on this region. Particularly, in blue are reported the photons acquired from silver, in green from zinc, and in red from oxygen. This analysis confirmed that the black particles correspond to the silver nanoparticles. The photocatalytic efficiency under UV light was tested by the degradation of 3 different organic pollutants: methylene blue, paracetamol, and sodium dodecyl sulfate. SDS is often used in many cleaning products for its foaming effect, and consequently it is present in grey waters. In the figure, the percentage of removed pollutants after 4 hours of irradiation are reported. The 2 typologies of composites were tested: silver, zinc oxide PMMA, and zinc oxide PMMA. The blank solution, that is a solution of the pollutant in the ionized water without the photocatalyst, was always measured for comparison. It's worth to note that the materials enriched with the silver were always more effective in the degradation of all the 3 compounds. In particular, they are able to remove about the 90% of both paracetamol and SDS present in the solution. The antibacterial properties of the silver zinc oxide PMMA nanocomposites were evaluated by measuring the survival rate of Escherichia coli exposed to the samples, both under UV light irradiation and also in the dark for 1 hour, to estimate the contribution of the photocatalysis. PMMA did not reveal any antibacterial activity, neither in the dark nor under UV light as expected. In the presence of zinc oxide PMMA samples, the bacteria survival rate was reduced up to 60% in the dark and to 40% under UV light. This difference is due to the photocatalytic contribution. Instead, in the presence of silver, the bacteria survival rate was reduced to 0, both in the dark and under UV light, thanks to the intrinsic antibacterial properties of silver. For concluding, the main message that I would like to give you is that it is possible to play with the atomic layer deposition technique so to realize different hybrid materials effective in wastewater remediation. Before finishing, I wish to thank you for your attention, but also I would like to thank all the scientists who contributed to this research and the projects that supported this experimental study.
Many thanks, Dr. Impellizzeri, for very nice presentation. And the topic is very important because it's very important. The photocatalytic action in the wastewater treatment is one of the main tasks for advanced oxidation process, for example. Are there any questions?
Yes, I would have a question.
Yeah.
Eric Cerrato from the University of Cardiff. As first, thank you for the very interesting presentation, and zinc oxide was at the base of my PhD study. Then I have, I have to say, bond to the zinc oxide matrix. My question is, do you have any idea about the role of the matrix, of the polymer matrix, in the improvement of critical activity? Thank you very much.
Okay.
In all the studies that I presented, the polymeric materials have as the role of substrate. So in this case, there is not a role of the polymers in the photocatalytic performance of the materials.
Thank you very much.
You are welcome.
Okay. One more question? Okay, I prefer to continue the section, and at the end, if we have time, we will continue. So we will continue with, I don't know, much more question. Okay, thank you very much, Dr. Impellizzeri. So we are going for the third keynote speaker, Professor Evrem Curcio. Professor Curcio is a chemical engineer and full professor in chemistry and chemical technology in the Department of Environmental Engineering at Calabria University. He obtained a PhD in chemical engineering and materials in 2005, carrying out research related to membrane crystallizers. I would like to mention in 2014, he was visiting scientist in the Department of Chemical Engineering to MIT in Boston, working with the research group of Professor Myerson. So just to conclude this presentation, his main expertise is in the activity related to membrane process and material science. And the title of his presentation is Membrane Technology at Water Energy Raw Material Nexus. Okay, please, Professor Curcio.
A sustainable management of water, energy and raw materials plays a central role in the Agenda 2030 of United Nations and in the New Horizon Europe program. Water Energy Raw Materials Nexus emphasizes their interconnectivity and represents an innovative approach both to reduce conflicts in the allocation of resources and to increase the resilience towards climate change. In this presentation, I will discuss on potentialities of membrane technology with specific focus on desalination, in developing possible solutions where the use of a nexus approach can lead to improved outcomes in integrated and sustainable management of strategically relevant resources. Within the next 20 years, many regions of the world will undergo water stress due to the increase of population, rapid industrial development in emerging countries, climate change, progressive desertification, and anthropic contamination of water bodies. With more than 18,000 of industrial installations operating worldwide, about 8 million cubic meters per day of drinking water produced, more than $15 billion of market value growing at annual rate of 9%, desalination is today the most reliable technological answer to the increasing demand of water. Desalination industry is dominated by membrane technology. Reverse osmosis accounts for more than 60% of the global capacity and almost totality of new contracted plants. Despite this success, membrane desalination still faces some critical issues. Water recovery factor is limited to about 50% as a consequence of osmotic phenomena and concentration polarization. This means that for each cubic meter of seawater treated by reverse osmosis, around a half cubic meter of the salted water and a half cubic meter of concentrate, usually referred as brine, are produced. According to the current practice in desalination, brine at salinity of about 70 grams per kilogram and temperature around 35 Celsius degree is directly disposed back to the sea. However, many studies in literature confirm the significant ecological and toxicological impact of hypersaline brine on marine flora and fauna. On the other hand, it's worth consider that brine contains about 3.5% of dissolved salts with prevalence of sodium and chloride ions, significant presence of magnesium, listed as critical raw material by European Union, and other trace elements potentially extractable such as lithium, rubidium, cobalt, molybdenum, and so on. While availability of raw materials is crucial for a sustainable industrial development, European Union is strongly dependent on the import of several metal ores from third countries, with significant implications in terms of market variability and availability fluctuations. This is a relevant weakness in European Union policy. On June 2020, we started the C4Value project, funded within Horizon 2020 program and coordinated by EURECAT, aiming at developing radical innovations to recover minerals and metals from seawater desalination brine. Target elements are magnesium, boron, and trace metals such as lithium, scandium, gallium, vanadium, indium, rubidium, and molybdenum. The extraction of trace minerals is facilitated if it occurs from highly concentrated solutions. Membrane distillation, operated as a standalone unit or downstream reverse osmosis stage, is an emerging hybrid thermal membrane operation driven by a temperature gradient having the potential to reach up to 85–90% water recovery, coherently with zero liquid discharge paradigm. As opposed to conventional pressure-driven membrane operations, in membrane distillation there is no filtration or transport in liquid phase. Water evaporation from seawater takes place at the interface of a microporous hydrophobic membrane. Here, water vapor diffuses across the porous membrane, while non-volatile components, such as dissolved salts, are totally rejected. Low operative temperature difference, around 30–40 Celsius degree, allow a feasible distillate flux in the order of 1–5 liters per square meters per hour, offering the possibility to exploit renewable energy sources such as solar, geothermal, and so on. Experimental investigations also demonstrated the possibility to dehydrate brine up to supersaturation for some dissolved salts, such as sodium chloride. On the left, a picture of typical cubic crystals nucleating on the surface of a membrane. This opened the way to the innovative concept of membrane crystallization. Application of membrane distillation at large scale is hindered by a relatively low thermal efficiency due to temperature polarization. This phenomenon refers to the decrease of feed temperature at the membrane interface, and it is intrinsically related to the latent heat flux of the evaporating water. As a result, the driving force to mass transfer decreases, and process performance are deteriorated. To overcome this limitation, in 2016 we developed the concept of photothermal membrane distillation by exploiting the ability of metal nanoparticles to efficiently release heat under optical excitation. In a pioneering study, we incorporated silver nanoparticles in polyvinylidene fluoride membranes, thus promoting locally the generation of heat under UV radiation. Experimental tests on both pure water and 0.5 molar sodium chloride solution, mimicking seawater salinity, carried out using membranes with different silver nanoparticles loads and irradiated at wavelength of 366 nanometers, resulted in water vapor fluxes up to 9 and 11-fold higher than the corresponding values for unloaded PVDF membranes. In order to mitigate limitations related to the high cost of precious metal nanoparticles and to the high energy demand of ultraviolet radiation, we have also checked the feasibility of carbon-based materials under solar illumination. For instance, infrared camera images you see in this slide show that membranes functionalized with vertically aligned graphene under sunlight exposure exhibited a significant increase of interface temperature up to 50 Celsius degree. In general, energy management is a crucial aspect in desalination. Starting from the pioneering investigations of Loeb and Surirayan, who fabricated the first asymmetric membranes made of cellulose acetate in the late 70s, the specific energy consumption of seawater reverse osmosis systems decreased from more than 12 kWh per cubic meter of desalted water down to the current 3-4 kWh per cubic meter. This important achievement, that is the main reason of the success of membrane technology over traditional evaporative processes, was driven by groundbreaking innovations in materials, with the introduction of thin film composite membranes, and in processes, with the use of energy recovery devices. However, energy consumption is still far from the thermodynamic value of 1.1 kWh per cubic meter predicted for 0.5 molar sodium chloride solution at 50% recovery factor. The energy of a pressurized brine is today recovered as electricity in Pelton turbines, or used to pressurize the feed stream by pressure exchangers. In addition, highly concentrated brines possess a significant chemical potential in the form of Gibbs-free energy of mixing that can be converted in salinity gradient power, a promising source of renewable energy usually referred as blue energy. A theoretically available amount of energy generated by mixing 1 cubic meter of diluted and 1 cubic meter of concentrated sodium chloride solutions is in the order of kilojoules, depending on the extent of the concentration gradient. Taking into account the flow rate and the average salinity of major rivers flowing into seas and oceans, a global potential of about 2,000 terawatt-hour per year has been estimated. Together with pressure-retarded osmosis, reverse electrodialysis is one of the best assessed membrane technology to harvest salinity gradient power. Our interest in this technology originated from the participation to the project REPower, funded by European Union within the 7th Framework Programme. In reverse electrodialysis, cation exchange membranes having negatively fixed charges in their polymeric structure, thus being able to repulse anions and to transfer cations, along with anion exchange membranes, mutatis mutandis, are alternatively stacked into a plate and frame module. Spacers interposed between each membrane couple form adjacent compartments fed with low and high salinity solutions. Under a concentration gradient, ions with opposite charges diffuse in opposite directions. This segregation of charges is converted at the electrodes in an electric potential difference, referred as open circuit voltage, theoretically defined by Nernst-Planck equation, where alpha is the perm selectivity of ion exchange membranes and gamma is the activity coefficient of ions in solutions. Recently, we designed a hybrid system integrating reverse osmosis, membrane distillation, and reverse electrodialysis to meet simultaneous production of water and generation of energy. Reverse osmosis retentate stream, with typical concentration of 1 molar sodium chloride, is processed by membrane distillation operated at recovery factor greater than 80%, so obtaining a hypersaline brine. The latter is fed to a reverse electrodialysis unit and mixed to seawater for salinity gradient power production. Experimental measurements on a red stack assembled with commercial Fujifilm membranes and working as a sort of seawater battery showed the possibility to extract a maximum gross power density in the range of 2.5 watt per square meter membrane per surface. A promising result but still not sufficient for practical applications. Interestingly, tests carried out at higher temperature resulted in enhanced performance due to improved ion mobility and reduced ohmic resistances. For instance, when mixing 0.5 molar and 5 molar sodium chloride solutions, a 50% increase in gross power density extracted was observed when warming up feed temperatures from 20 to 50 Celsius degree. This evidence paved the way to the conversion of waste heat into electric power. Practical implementation of RED strongly depends on the development of a new generation of ion exchange membranes with improved perm selectivity in a hypersaline environment and with low electrical resistance. Electrochemical impedance spectroscopy analysis demonstrated that the extent of membrane resistance largely prevails on both electrical double layer and diffusion boundary layer resistances. In addition, the negative impact on the resistance of cation exchange membranes of multivalent ions and in particular of magnesium, the second most abundant cation in seawater, drastically reduces the gross power density that can be extracted when operating with natural feeds. This slide summarizes our vision on the future of membrane desalination in the frame of a circular economy. We propose a radical paradigm shift where brine is no longer considered an harmful waste to dispose, but as a potential source of water, coherently with a strategy of process intensification, of renewable energy in the form of salinity gradient power, and as an alternative and potentially unlimited mine for the extraction of valuable minerals, most of them not present in Europe, but having a strategic relevance for a long-term sustainable development of our society. In order to increase the reliability of this approach, further advancements are needed. Efficient integration of waste heat and membrane distillation as necessary step towards high temperature reverse electrodialysis, development of new photothermal membranes with stable properties over time for efficient sunlight-driven evaporation, development of new ion exchange membranes with high perm selectivity, low electrical resistance, and low cost for practical applications. Thank you for your attention.
Many thanks, Professor Curcio. It's very interesting presentation. I had a lot of the integration, the possible integration of membrane with reverse electrodialysis, etc. So, are there any questions? So, I try today to make some one curiosity. You said that about 50% of recovery of water could be possible in the water treatment, especially from sea, in the reverse osmosis. Do you know which kind of activity are carried out to improve this possible solution? Only this your approach, or there are also other approaches to improve these steps?
Well, presently, membrane distillation is the most reliable approach to increase this kind of recovery factor, because reverse osmosis, that is the reference technology, is limited by osmotic phenomena maximum to 40-50%. Some limit of materials resistance doesn't allow to overcome this limit. So basically, membrane distillation and in general membrane contactors are presently the most reliable solution option for increasing the recovery factor of water.
No.
And the possibility to exploit the photothermal effect increases also the reliability in terms of energy management, of course, of this approach.
No, yeah. Okay. Many thanks.
Efrem, just a question. Can you comment on existing industrial plants of membrane distillation?
Well, at present, we have only pilot plants, even if big pilot plant, especially in Korea. Korea is investing a lot in this technology. And I have to say, well, the results are promising, because especially what is also has to be improved in membrane distillation is the long-term stability of membranes against wetting phenomena, for instance, generated by organic fouling. In this case, it has been observed that appropriate pretreatment systems that are also used in desalination can prevent this negative impact of fouling. Basically, the concept is to integrate membrane distillation in existing seawater reverse osmosis desalination plant in order to benefit about the pretreatment that are already implemented for this desalination system.
Okay. Are there any questions? Okay.
Can you comment on fouling?
Sorry.
Can you comment also on fouling and existing efforts to avoid the problem of fouling in desalination technology?
Well, in principle, battle against fouling is running since 50 years, so the beginning of membranes. Fouling basically is intrinsic to these operations, as corrosion is intrinsic to metallic materials. Let's say, again, very important as the pretreatment system, in particular, new membrane integration of membrane technology, and in particular microfiltration, ultrafiltration, to the removal of bacteria and bacteria or algae that are present in seawater, natural seawater, since biofouling is the more aggressive form of fouling that is usually irreversible. The possibility to incorporate, for instance, silver nanoparticles, as proposed in our approach, photothermal approach, can also help in reducing the biofouling.
Thank you very much.
Thank you very much. Are there any questions? Just last question. Okay. Okay, so this section is concluded, and I would like to thank to all the people, to all the participants, to this very interesting discussion, keynote presentation, etc. So I think now there is like a break, and I would like to give you my best regards and all the best for the continuation of the Congress. Thank you very much for everybody.
Good afternoon to everybody. It's time to restart this session 3 on advanced materials for water treatment and environmental sustainability. I just want to check if Despina is ready for his presentation, for her presentation.
Yes, I am here.
Okay, fine. So it's my great pleasure to present the next invited speech from Despina Fraguli from Italian Institute of Technology in Genova. And the title of this work is Porous Polymeric Composites for Water Oil Separation. Please.
Good afternoon. I'm going to share my screen. Okay. So, good afternoon to all. Today I'm going to talk to you about porous materials that we're developing in the Smart Materials Lab of IIT. And these porous materials are based on polymers and nanoparticles, and we use them for water oil separation. Water oil separation is somehow a very tricky task, and this is because oil can be present in water in different forms. It can be present as emulsified oil, as dispersed oil, or as free oil, and depending on the state, on the way that the oil is dispersed, is present into the continuous phase of water, different separation methods have to be adapted. Specifically for the free oil separation from water, we usually use sorbents, and these sorbents have to have very specific characteristics in order to have a successful water oil separation. Specifically, these materials have to be highly selective, so they have to absorb only the oil. They have to have high absorption and capacity, and also high rate of absorption. They have to retain the oil after the absorption process. They have to float on water, and also they can be reusable and cost effective. And this is, of course, in order to have more sustainable solutions in this process. Free oil can also be separated from using gravity-driven filters of filtration, as also the dispersed oil. And again, in these situations, the requirements are that we have to have materials that can have high oil rejection efficiencies. They should not cause any secondary pollution, and of course, a very very big plus in these materials is that they have to be reusable and cost-effective. Together with all these requirements, recently, the very recent trend in the scientific studies that have to do for water oil separation is also to develop materials that can separate in a single step not only the oil from the water, but also other additional pollutants, because of course oil is never, or in a very few situations, oil is the only pollutant of water. There are also other pollutants inside. In this talk, I'm going to talk to you about few examples of the works that we are doing. And these examples have to do with water oil separation, developing materials for water oil separation through sorption and through gravity-driven filtration. Going to the sorption process, what we do and what is also very used to be done in the current studies is to take already existing porous materials and try to change their surface properties in order to transform them into efficient oil sorbents. Specifically, we work with polyurethane foams that are developed for other reasons, for packaging, for furniture, for housing. And after the end of their regular life, we take them and we functionalize them, we change their properties through nanotechnology in order to make them oil sorbents. This work started a few years ago in the lab, and the first study that we did was dealing with the utilization of polyurethane foams, where we applied superparamagnetic iron oxide colloidal nanocrystals on their surface through an adsorption dipping process, and then a subsequent layer of Teflon nanoparticles was applied on as a second layer. The result was that the foams from the smooth morphology of their pores, they turned to have a very rough and very particular nano-rough surface, which was due to the presence, of course, of the nanoparticles. On the top, these materials had the ability to absorb the oil very fast, but at the same time, the water was completely going out from the polymer, from the foam. So the result was that this foam could float on the surface of the water, and since it was also magnetic due to the presence of the magnetic nanoparticles, we could easily move it towards the oil area, the oil spill, in order to remove the oil, and then we could recover it without any contact. This material and this study was somehow a small proof of concept, and after this a lot of other studies came out that were dealing with a similar idea. So to take already used, already formed foams, and then through a surface functionalization to transform them into selective and efficient oil sorbents. The results in all these studies were very nice, and also the functionalization processes were very interesting. But the thing that was missing from all these studies was what is the effect of the pristine foam on this process. What foam we should choose in the very beginning in order to functionalize it and in order to have a very impressive and very high performance for the oil absorption. And this is something that we try to do in the next study, to investigate the substrate, to investigate the pristine foam and the effect on the oil absorption. So what we did, it was that we chose polyurethane foams. We found polyurethane foams that had similar porosity, but their difference was on the pore structure. So there were polyurethane foams that had highly interconnected pore structure, medium interconnected pores, and low interconnected pores. And then they had also pores sizes that were different. And then we tried to see what is the effect of this structure on the oil absorption capacity of the material. So what we saw is that foams with highly interconnected pore structure and small pore sizes that are below 1,000 micrometer could absorb the maximum amount of oil. So choosing the right foam in the beginning could make the difference on the oil absorption process. Then the further functionalization, so the further surface functionalization with the nanocomposites that we're using, could affect and change the surface roughness and the surface chemistry of the foams, transform them in super hydrophobic and super oleophilic, so enhance significantly the selectivity of the oil absorption process, of the oil separation process, and also enhance the oil absorption rate. But we have to stress that the maximum oil absorption capacity always was dependent on the initial structure of the foam. And this we can see it here, where we have two foams that have different pore structure, highly interconnected and medium interconnected. We have the same functionalization process on their surface. And as you can see, the foams with high pore connectivity can reach oil absorption capacities up to 60 grams of oil per gram of foam, while the other has almost half of it. In both cases, I should say that the functionalization process was pretty stable. The process was selective, so we had enhanced selectivity, and the oil absorption capacity could be not altered a lot after several desorption cycles. Going to a next type of water oil separation, which is the gravity-driven filtration. We work with polymeric-based 3D filters to do this process. We choose polymers because with polymers we have a great choice, so we can choose the right polymer in order to have the appropriate chemistry, surface chemistry, and the properties that we need in order to form our material. We chose also polymers because with the polymers we can choose the fabrication process in order to make our porous system. So we can go from freeze drying to electrospinning, and we can in this way tune the pore sizes and the pore characteristics. Furthermore, with polymers, using polymers as support materials, we can combine them. Polymers can host very, very well and a very great variety of nanomaterials. So in this way, it's very easy to add functionalities on our systems. And with all these choices that we can have, so to choose the right polymer, to choose the right fabrication process, to choose the right composite, the right nanomaterial or the right molecules in order to make the further functionalization, we can arrive to the characteristics that we need for the gravity-driven filtration. And the characteristics that are mainly needed for this type of process is to have materials that can have underwater super oleophobic properties. So apart from all this, using also polymers, we can we can make 3D filters, so we can make foams, porous materials, and in this way we can induce more and longer interconnect permeation channels in our final materials. So in the first example, I show you here how we combine waterborne polyurethane with expanded graphite flakes and calcium carbonate, which is used as a porogen. And by a simple mixing without the addition of any additional solvent, we can form finally a foam. And this foam is a composite foam that has a dual porosity, dual pore sizes. As you can see here in the scanning electron microscopy and the morphology of the foams, we have we have different, very particular pore structure. And from mercury intrusion porosimetry, we see that we have pores, big pores that are around in the range of few hundreds of micrometers, and these are attributed to the how the polymer sticks together the worm-like macrostructures of the expanded graphite, and smaller pores of few micrometers that are attributed to the presence and then the subsequent removal of the salt that we use as a porogen in the whole process. This type of material, of porous material, is of course water absorbing. You see that the water droplet is absorbed very fast from the surface, and its underwater oil contact angle is very high. So underwater, you see, using different types of oily substances, in all cases we have an underwater oil contact angle which is above 140 degrees, and the sliding angle very low, in most of the cases below 10 degrees. So this material is underwater oleophobic, and we further go and see how this material, and if this material is good for a gravity-driven filtration of an oil-water mixture. As we see here in this graph, in all the flow rates that we use, and when I say flow rates, this is defined actually by the volume of the feed solution that we use, because we have a gravity-driven filtration. So in all the volumes of the feed solution, we have an excellent oil rejection efficiency, which is above 99.98%, so almost 100%. And here you see the experiment, how it is. So we have the mixture and we pass it through the filter and then we analyze the water. And this oil rejection efficiency is stable over different filtration cycles. So the foams can be reused and without losing any performance. On the top, this type of foams are very robust. And you can see it here that we subject the foam in mechanical abrasion cycles. And in all the cases, up to 25 abrasion cycles that we have tested, the oil rejection efficiency is not changing. While at the same time, the foams can work also in corrosive environments, so they can separate efficiently oil from water in the case that the water has different acidities, or also in the case of that the water is seawater. Finally, the last example is dealing with the formation of PDMS-based foams for water oil separation. We form PDMS foams with interconnected pore structures, with hydrophilic and underwater superolophobic properties, and also with antibacterial properties. And in this way, we can reach to an effective oil-water separation, but also a simultaneous oil sanification of the permeate. This we do it by first fabricating the PDMS foam following the high internal phase emulsion process. Actually, we mix PDMS with water and solvents and other components, and more details on this fabrication process you can find in the related publication. After the curing, we have the PDMS foam that looks pretty nice, pretty robust, and as in the photo. This foam doesn't have the appropriate surface properties, of course, because it's a pure PDMS foam that has, and therefore it's hydrophobic. Therefore, what we do is we coat the pores of the foam with polydopamine through a dipping process into a dopamine solution. And then subsequently, in order to add further functionalities, we dip the polydopamine coated foam into silver ion solutions. And this has as a result the formation of silver particles on the surface of these types of foams. As you possibly saw also on Dr. Athanasiou's publication, the morphology of the PDMS foam is very nice and it has highly interconnected pores. And this pore structure is not affected by the fabrication process. But the morphology of the foam, so the pores are decorated after the fabrication process with silver nanoparticles. This silver nanoparticles density depends on the initial silver nitrate concentration that we use for the dipping, and therefore in this way we can somehow control the density of the silver nanoparticles onto our material. We can also control the wetting properties with this situation, with this fabrication process, because as you see here, the more are the silver particles on the surface of the foam, the more is the underwater oleophobicity of the foam, the higher it is, reaching to a situation in the higher amount of silver where we have a super oleophobic foam underwater. And also we can control the flow rate with this fabrication process because, again, depending on the amount of silver particles onto the material, we can have flow rates that are slower and they are becoming faster as the material becomes more underwater superolophobic and also due to the presence of the more silver. On the other hand, the oil rejection efficiency in all cases is pretty stable and very high. Then if we go to analyze the permeate after the filtration process, we see that the silver ions that are released into the permeate during the filtration are more in the case of the slower flow rate. And this is something that we would expect because the water is in more time in contact with the silver nanoparticles in order to remove somehow the ions. And in the case with the more silver ions, we see that the permeate has antibacterial properties. Therefore, as a last step in this study, we use as a feed solution oil, water, and bacteria all together in a mixture, and then we try to make the filtration and analyze what is happening. What we see is that the oil rejection efficiency is not affected by the presence of the bacteria in the feed solution. So we always have very high oil rejection efficiency. And also, but the thing that we see is that the antibacterial performance, so the permeate does not permit to the bacteria to grow. So it has antibacterial properties. Therefore, we could successfully separate oil from water, but at the same step also to sanitize the water. With this, I would like to close. I would like to thank my colleagues that worked in this project, and also thank you all for your attention.
Thank you, Despina, for your very nice presentation and convincing approach. Okay, the floor is open for the discussion. Any question from the audience? If not, or waiting for someone that want to make a question. I have a curiosity. Is it possible to use similar materials and similar approach in producing oil and water emulsion? I mean, also from industrial point of view, the production of emulsions is very, very important in many applications.
Okay, you mean that to try to produce oil, water, and this, in principle, is something that we somehow indirectly investigated when we're trying to make the PDMS foams. So we were trying to make a stable emulsion of an oily substance into the water. And this was a very difficult condition. We made a lot of trials in order to find the right compositions of the oily solvent, which was the oil solvent of the PDMS of the water. And usually in order to make it, we had to use some surfactants, but also the presence of some nanoparticles, like the silicon dioxide nanoparticles that we used in order to form this type of emulsion was very important. So in principle, yes, this could be done by finding the right composition, let's say, on the materials.
Yes, but it's very interesting from my point of view. It's a very nice approach that can be, let's say, a double way, no? Removing oil, but also producing emulsion.
Yes.
Can be interesting. I don't know if any question from the audience. If not, let's thank once again Despina, and we move to the second invited lecture that will be given by Sabrina Carroccio from Polymers, Composites and Biomaterials Institute of the National Research Council of Italy, located in Catania. The title of this presentation is Novel Polymeric Resins for the Removal of Organic and Inorganic Contaminants from Water. Please, Sabrina.
Good morning, everyone. Thank you for joining us today. Since my colleagues have put you up to speed on social challenge to be faced to ensure clean water in the next future, I'm going to make use of the time left to present you an overview on acrylate-based materials produced in our lab for the removal of toxic organic and inorganic contaminants. Specifically, I'm going to be talking about the formulation of polymer acrylates material in form of cryogel, able to mineralize organic pollutants by absorption and subsequent photocatalysis, but also properly addressed to chelate toxic metal ion. Finally, molecular imprinting polymer technology can be also ad hoc exploited to selectively absorb emerging pollutants and particularly drugs. Let's start by introducing the design of hybrid polymeric materials for the photocatalytic degradation of organic pollutants. As active support, we have selected poly-2-hydroxyethylmethacrylate, polymerizing it via cryosynthesis. The latter is a traditional radical polymerization at temperature below the freezing point of the solvent used, in our case water. In such conditions, macroporous absorbent materials with high toughness and superfast responsivity were obtained. In brief, the polymerization takes place in the presence of cross-linking agent, forming a network around the ice crystals that act as a porogen. When the system is defrosted, the ice crystal melt, leaving interconnected macropores having shape and dimension complementary to them. Looking into the synthetic procedure here reported, we performed the polymerization by ammonium persulfate as initiator at temperature of -16 degrees in the presence of methylene bisacrylamide as a cross-linking agent. To enhance the sponge absorption toward the pollutants, we also added graphene oxide as a filler during the synthesis, obtaining a black sponge reported here in the picture. While absorption aptitudes were provided, now we have to add the ability to degrade the absorbed pollutants. And to do this, both materials with and without graphene oxide were subjected to zinc oxide functionalization by atomic layer deposition. Notably, taking advantages from the hydroxyl pentyl group of the polymer, our strategy also involves the formation of stable covalent linkage between the polymeric support and the nanostructured zinc oxide molecules, avoiding nanoparticles release during the purification process. The produced materials were characterized by using chemical, physical, and morphological analysis. Thermal gravimetric analysis was used to provide quantitative data on zinc oxide deposition, whereas XRD confirmed the formation of wurtzite, which is the photoactive crystal form. The scanning electron microscopy reveals the typical morphology of zinc oxide formed by small grains uniformly distributed on the surface, showing a coating of regular shape and size with a grain average diameter of 10 nanometers and a thickness of 15 nanometers, as indicated by TEM. All data collected were correlated to the photocatalytic results. To this end, sponges with and without zinc oxide were immersed into a methylene blue solution at no concentration, and their absorption were measured up to 5 hours. After that, the sponges were extracted from the dye solution and irradiated by UV light. The results are visible to the naked eyes. Samples containing zinc oxide discolored since photocatalytic degradation of the blue dye occurred. As it turned out, materials were regenerated and can be reused for several absorbing and degrading cycles. Of course, these outcomes were confirmed by FTIR analysis. If you are interested, more detail in the paper here reported. The second approach is aimed to tackle water pollution from emerging contaminants derived from the use of pharmaceutical and personal care products. As you can see from the table here reported, their accumulation was already seen in the water bodies. It is important to point out that even if the traces of emerging pollutants are minimal, they can cause serious threat to the human health. The challenge we face here is the making of a material which, although in the presence of other contaminants, can selectively remove only the target molecules, keeping it from reaching the resin saturation and losing efficiency in capturing the target drugs. The approach used benefits from molecularly imprinted polymer technology. To this end, methacrylic acid is polymerized and cross-linked in the presence of the molecules that we want to eliminate from water. The monomers are selected for their ability to form chemical interactions with the template molecules. After the synthesis, the template is removed by washing the material with an acidic solution, leading to the formation of sites having form and type of interaction able to target the drug, which is, in our case, diclofenac. The whole process of synthesis and absorption was tested by pyrolysis GCMS analysis. From the profiles, it is possible to appreciate the signals of diclofenac before washing, its disappearance after washing, and again its appearance after absorption process. Quantitative data obtained by UV spectroscopy showed as the formulated system can provide high absorption capacity with high selectivity, and plus it can be reused and regenerated up to 4 cycles. Finally, I would like to show you the design of novel cryogels containing M-methyl-D-glucamine as active group. The macroporous materials were prepared via sustainable cryopolymerization by using as comonomers vinyl benzyl M-methyl-D-glucamine and 2-hydroxyethyl methacrylate at different percentages. Particularly, cryogel containing the maximum percentage of glucamine monomer showed excellent chelating ability versus arsenic, chromium, and boron metal ions. If compared with similar materials, the macroporous architecture makes the difference. Indeed, their interconnected nature provides fast diffusion pathway, as shown in the video, determining exhaustive access to the chelating sites, thus boosting the ion sequestration as revealed from the kinetic profile. The cryogels were indeed characterized. In particular, scanning electron microscopy reveals different pore sites depending on the percentage of comonomer used, decreasing by adding more to hydroxyethyl methacrylate. Compared to nanoparticles whose excellent absorption properties arise from the high surface area, freestanding cryogels result safely and manageable to be used. Also, additional benefits consist in their lower flow resistance and filtering capacity of heterogeneous solutions. The materials are able to drastically reduce the ion content in a few minutes within the compliant regulations. Besides, as evident in the video, it can be used as a sponge, providing an interesting adding value. To make visible the absorption process, we used a solution of chromated ions being yellow with a huge concentration, more than 1000 ppm. After solution uptake, the purified medium can be released by simple switching the sponge while contaminants remain entrapped inside. To simulate real conditions, the interference of competitive ions was also tested by ICP-MS, adding up to 3 different salts in water. It can be observed that the presence of phosphate did not affect both arsenic and chromium sorption processes, whereas they are influenced by the sulfate, but only for concentration higher than 16 ppm. It was also demonstrated that the has prepared materials can be reused up to 6 cycles without depletion of performance and regenerated up to 3 cycles by washing with an acidic solution. In the video, the resin is treated with an acidic solution. Of course, in this case, the high amount of metal oxygen absorbed needs more washing procedure to achieve a complete desorption. To sum up, I have reported some relevant proof of concept to employ in the field of water remediation. We have produced reusable materials that can absorb the organic contaminants, degrading them by exposure to UV light. In this view, we are working now to develop photocatalytic hybrid system to work under solar irradiation. In addition, the design of a low-cost system to selective confined drug molecules was performed. Finally, chelating resins to remove metal ions were also reported. It is clearly proven that chelating capacities of our materials is far higher if compared to materials reported in the recent literature and also those present in the market. In light of this, we have applied for a patent for which we have recently received a positive evaluation from the European Patent Office, as well as positive feedback from companies interested in the invention. Furthermore, the patented material, namely Cryopura, was financed by the Ministry of Economic Development to increase the TRL from 4 to 7. To this purpose, we are working towards the development of demonstrator systems, scaling up the material production. The last but not least, Cryopura has been selected to take part in EIT Jumpstarted 21 competition and to compete for CNR to IP Award 2021. I'd like to thank all people that worked and keep working in this project, especially the whole CNR team, the organizers and the scientific committee that invited me to present this work, and of course all you for your attention.
Thank you so much, Sabrina. A very nice presentation. Also, really appreciated the scale-up efforts that make that transfer the technology from bench scale, laboratory scale to let's say industrial application. Never easy, but it's important to try to do this. Is there any question from the audience? Please.
So good afternoon, and thank you so much for your interesting presentation, rich of very useful outcomes. My name is Sergio Santoro, and I've been always curious about molecular imprinting, and it's a very elegant method to recognize molecules in a very diluted solution. But do you have any information about the long-term stability of the imprinting?
No, we are working on because it's a very difficult task to achieve with a lot of trials to demonstrate the stability along the time. But good is a good question. Up to now, we have performed up to 6 cycles of regeneration, and it works. Yes, it works.
Thank you. We have, I think, a couple of minutes for at least one more question. And thank you, Sabrina, for keeping the time. Just out of curiosity, I saw also the possibility or the goal to remove arsenic or other harmful contaminant. let's say, after it's no longer possible to regenerate your cryogel, how you manage, I mean, your waste? I mean, what to do at the end of the process?
Okay, so it's a question that toward the sustainability of the materials, of course. There are studies to depolymerize the cryogel, because the cryogel is based on a polymethacrylate that is well known that can be depolymerized by increasing the temperature up to the decomposition temperature. And Arkema is a leader of polymethacrylate production. Has at the moment a European project to do this. So we hope in this study.
Yeah, so the polymerization of the...
Depolymerization, and so the use of the polymerized monomers to polymerize again.
Okay, thank you. I think, yeah, it's time to move. Let's thank again Sabrina, and it's time to move to the last invited lecture that will be given by Domenico Caputo from University of Napoli, Federico II. The title is Advances in Nanoporous Materials for Water Treatment. Please, Domenico. You have a live presentation, or is registered? Can you switch on your microphone?
The microphone.
Okay. No, we don't hear you. We have, I think, a technical problem with your microphone.
Let's switch to the next presentation. In the meantime, we can solve this problem.
Okay, yes, good suggestion. So if Eric is ready, Eric Cerrato from Cardiff University.
Yes, here I am.
Okay, so sorry for anticipating your lecture, just for
no
yes,
problem.
So, thank you so much. Great. So Eric Cerrato is from Cardiff University and will present work titled Origin of Visible Light Photodegradation Ability of cerium oxide and zinc oxide heterojunction. Okay, please, Eric.
Yes, thank you very much for the kind introduction. Okay, may you see my presentation?
Yes. If you...
Okay.
Okay, perfect.
Oh, sorry, it's going... Okay, here.
Thank you again for your fast reaction.
Oh, don't worry, it's okay. As first, I would like to thank the organization committee that gave me the opportunity to have this talk about the origin of the photoactivity of the cerium dioxide zinc oxide heterojunction. Before to start with the characterization of comes about the mixed system, I would like to give you a couple of words about the transition metal oxide heterogeneous photocatalysis. In general, in a photocatalytic process, a substance, the photocatalyst, is activated by absorbing light. Light harvesting induces phenomena that increase the rate of a given chemical reaction without the consumption of the photocatalyst itself. Nowadays, the most studied and promising materials as heterogeneous photocatalysts are web and gap semiconductors, among which zinc oxide and titanium dioxide have shown the best performances. As reported here in the sketch, when a semiconducting material is irradiated with a suitable amount of energy, it can give rise to charge carrier separation. namely the photoexcitation of electrons from the valence band to the conduction band with the consequent formation of holes in the valence band. Then the electrons in the conduction band and the holes in the valence band are directly the actors of reductive and oxidative redox reactions, respectively. In particular, in the topic for the topic of this session, heterogeneous photocatalysis is one of the most promising technology for the production of the advanced oxidation processes. That can allow the complete mineralization of a great variant of classical and emerging organic pollutants in wastewaters. Indeed, once at the surface, photoexcited holes can give rise to hydroxyl radicals, very well known as the strongest and not selective oxidant species. On the other hand, the photoinduced electron in the conduction band form superoxide anion that can be converted in peroxo species and still contribute to the photodegradation process. Here I reported the photodegradation of A-sulfate K, which is an emerging pollutant, and it is interesting to observe the photodegradation ability of our mixed system, cerium dioxide and zinc oxide, in comparison with the very well-known P25 in blue line in this slide. While upon UV radiation, the behavior of the two materials is almost the same, upon visible light, the performance of the mixed material in the photodegradation of the pollutants overcomes that of P25. Then the goal of our study was trying to understand the working mechanism behind this photoactivity. And this is a fundamental aspect in order to develop even more efficient photocatalyst. Then, from a structural characterization, XRD analysis reveals that the diffraction pattern of the mixed sample labeled as CZ1 is strictly similar to that of bare zinc oxide, except for a weak, barely appreciable reflection due to the presence of ceria. The presence of ceria also induces a slightly broadening of the XRD reflection, indicating smaller crystallized sites. These assumptions were confirmed by TEM micrograph, where here I reported the image of the mixed system, in which it is evidenced the presence of small ceria particles, mostly with quasi-spherical shape, stabilized on the zinc oxide surface. Then, summarizing from a structural and morphological analysis, it comes out that cerium doesn't enter in the zinc oxide lattice, forming extrinsic defects. Rather, there is a formation of small ceria particles anchored on zinc oxide surface, and implying the existence of the cerium dioxide zinc oxide interfaces or heterojunction. The UV visible DRS spectroscopic analysis evidenced that for the mixed sample is characterized by is dominated by the valence band conduction band optical transition of zinc oxide. But in addition, there is a further absorption around 400 nanometer with a tail in the visible region. The presence of ceria in the mixed sample doesn't affect the band gap of the material. Then EPR spectroscopy study was crucial in the determination and in the rationalization of the working mechanism of the mixed system upon visible irradiation. Indeed, EPR spectroscopy is a very powerful technique in order to follow charge current separation in such modified semiconductors. In addition, it is possible to perform a pre-screening of the material photoactivity. Here in panel A, I reported the EPR spectra recorded upon different wavelength irradiations for the mixed system. And we can observe that already before irradiation, the material shows some EPR signals deriving from the lack of stoichiometry of the synthesized sample. Upon different wavelength irradiation, it is possible to observe the growth of the symmetric line at G1 point in 96 due to the stabilization of photoexcited electrons in zinc interstitial plus species, inducing Deffel levels just below the conduction band edge. This phenomenon is accompanied by the parallel growth of the signal D2O- in zinc oxide and arising from the stabilization of photoinduced holes. No signals due to cerium 3+ were recorded, and the highest charge carrier separation was recorded for a cutoff filter at 420 nanometer. Focusing our attention on panel B, where the double integration of DPR signals for trapped holes and trapped electrons are reported, we can observe that the amount of trapped holes is systematically higher than that of trapped electrons. And this is not the case for bare zinc oxide. Then this peculiar behavior of the mixed system shed the light about the working mechanism upon irradiation. Indeed, also helped by a theoretical calculation performed in collaboration with the Professor Pacchioni group in Milano Bicocca, we were able to identify an electronic transition from zinc oxide conduction band to the empty localized for F orbitals of cerium 4+, which was reduced to cerium 3+. Cerium 3+ is a paramagnetic species, but due to its high spin-orbit coupling constant, cannot be measured in this kind of measurements. Then, a part of photoexcited electrons are stabilized as cerium 3+ and cannot be detected by EPR. Still, explaining the different amount of trapped holes and trapped electrons that we recover for the double integration of DPR signals. Still, from the theoretical calculation, it appears that a peculiar atomic arrangement occurs at the interface of the two oxides, inducing a band gap reduction at the interface of the two materials. Generally, in a photocatalyst, it is not just important to observe a charge carrier separation, but also to verify that the photoinduced charge carriers are present at the surface of the material in order to entail redox chemical reactions. Then, in this case, we performed the visible irradiation in oxygen atmosphere, and what we recorded was the typical signals of O2- or zinc 2+ and cerium 4+, indicating that the photo-induced electrons excited by visible photons were available at the surface for reductive chemical reactions. On the other hand, we also tested the availability of photoexcited holes at the surface of the material performing the irradiation in hydrogen atmosphere, since hydrogen is a hole scavenger. We can compare the irradiation performed in vacuum with the whole signal of the irradiation performed in hydrogen atmosphere, where the signal of stabilized holes is dramatically reduced due to their reaction with the hydrogen of the environment. Then we can still affirm that the photoinduced holes excited by visible photons are available at the surface for oxidative redox reaction. We tested the photocatalytic activity of the material also irradiating with visible light aqueous suspension of a DMPO molecule. DMPO can form a stable paramagnetic adduct with OH radicals that can then be APR detectable. OH radicals, as said in the introduction of the presentation, can be formed by the photoexcited holes by the material reacting with water molecules. In panel A, I reported the typical 4-line EPR spectra of DMPO OH adduct for bare zinc oxide and the mixed material. We can appreciate how the mixed sample can produce a very high amount of OH radicals in respect to the bare material, which is almost inactive. Then, concluding, cerium does not form extrinsic defects entering inside the zinc oxide lattice. Rather, there is a formation of a colony of small Syria particles anchored to zinc oxide surface, giving rise to a biphasic solid or heterojunction.
The mixed material has exhibited a higher charge carrier separation upon visible irradiation respect to bare zinc oxide, and we also tested the availability of the photoinduced electrons and holes at the interface, both at the solid solid gas interfaces. Finally, the theoretical model, supported by experimental results, help us to identify how the mixed system works under visible irradiation. Then, with the constitution of the cerium dioxide zinc oxide heterojunction, the electron-hole pair separation is improved, preventing the recombination. I would like to thank all the people that collaborated in this work, and especially the Professor Maria Cristina Paganini, that was my supervisor in the PhD at the University of Turin, and Professor Elio Giamello, and all of you for your kind attention.
Thank you so much, Eric, for the very, very nice presentation. Thank you for keeping the time. And okay, I open the floor for the discussion. Any question from the public? It's necessary to stimulate the discussion. But well, it's not easy in a virtual conference. This is what usually happens. Anyway, I have a curiosity concerning the EPR analysis.
Yes.
In particular, how to manage the effect, possible effect of polarization, I mean concentration polarization in the diffusing boundary layer close to interface, that for sure can affect the reaction kinetics that you are going to observe. I mean, components, reactants, products can diffusing back from the surface. I mean, how sensitive is this EPR analysis? Is EPR analysis to this situation?
Okay, EPR analysis in general is very sensible, also in detecting a very low amount of paramagnetic centers. The problem with this kind of system is the fact that we didn't have a hyperfine interaction. Then we were not able to how to say about you asked about the polarization. We didn't have a phenomenon that we can use in order to deeply study the materials, since without having hyperfine interaction, we could not evaluate the polarization, for example, as you said, at the interface of cerium dioxide zinc oxide. This is derived from the fact that zinc doesn't have a nuclear spin, or better, the isotope is too much low abundant, while for the cerium T+, as explained, the spin orbit Capri constant doesn't allow also the detection of the paramagnetic signal in the CW in the ECW measurement. Then we don't have an echo in the pulse EPR. Then we didn't have the opportunity and the choice to analyze the polarization at the interface of the two oxides.
And so how easy is to compare, for instance, measurements, EPR measurements with reaction data, experimental reaction data that I assume, I don't know, you perform in a rotating disk or in such kind of well-mixed reactors. So if polarization phenomena are neglected, I mean, in which range of fluid dynamics you are able to compare experimental, theoretical analysis, and EPR measurements?
Indeed, this kind of measurement.
You have some protocol, some clear. Yes, how to.
Yeah, these indeed these measurements are just pre-screening of the material photoactivity. Basically, in this case, we did the work. The promising photoactivity of the material was already new. The point was to investigate the working mechanism, why this material work upon visible irradiation. Then in this case, it was easier to define a tailor-made approach in order to to follow the charge carrier separation at the interface. Since we were interested to observe, for example, where are stabilized the photoinduced charge carriers. As I said, the most of electrons are stabilized as cerium 3+ that we cannot record in the mixed material. Also, in order to follow the electrons and the holes photoinduced, as I explained in the presentation, we adopted a different kind of irradiation in different environments. The point is that we can, this is due in a clean atmosphere. Of course, in this kind of measurements, we are not taking into consideration for example, the effect of air or of other, for example, pollutant agents inside that could be present in a real photocatalytic reaction. While for the last measurements about the spin trap measurements, the radiation of the aqueous solution of the MPO, in that case, you are at ambient pressure and ambient temperature. Then it is a more realistic evaluation of the OH radicals produced by the material. Then I would say that the real the determining measurements that it is more next to the real experimental condition of a photocatalytic process is the measurement of the OH radicals in solution.
Thank you, Eric. Very clear answer.
Thank you for asking.
I don't know if there is any question. Any other question? If not, I see from the chat that we are able to recover Domenico for his presentation. It's true?
I'm here.
Okay, fine.
Oh, good afternoon to everyone.
Again, Eric, thank you so much.
Thank you.
And
thank you,
we have Domenico with us. Great.
Yes.
Solving. I think that I already introduced you. So from University of Napoli, Federico II. The title is Advances in Nanoporous Materials for Water Treatment. So the floor is yours, please.
Thank you, Efrem. Thank you, Eric, for anticipating your presentation. Good afternoon to everyone. I try to share the presentation. Okay, I hope that you can see. Okay. Oh, thank you. My presentation regard advances in nanoporous materials for water treatment. As is well known, the International Union of Pure and Applied Chemistry classify porous materials in three categories: microporous, mesoporous, and macroporous, depending on the pore size. The term nanoporous materials has been used for those porous materials with the pore diameter of less than 100 nanometer. Many kind of crystalline and amorphous nanoporous material, such as tactile silicates, pillared clays, metal, and others, are of great technological interest. The main application field of nanoporous materials include catalysis, molecular sieving, energy, environmental protection, and more recent healthcare technologies. Since the middle of the last century, zeolites have been the class of nanoporous materials of greatest technological interest. They are microporous crystalline aluminosilicates having a three-dimensional structure based on silicon and alumina-centered tetrahedra linked by corner and framework are usually very open and contain channels and cavities. Each aluminium atom introduce one negative charge, and this charge have to be neutralized by a certain number of cations naturally surrounded by hydration water molecules. The great technological interest of zeolites is due to their valuable physico-chemical properties such as cation exchange, absorption, molecular sieving, and catalysis. Natural zeolites, for instance, have found extensive application in environmental properties, including water, wastewater treatment. In fact, exploiting their properties, especially the high cation exchange capacity, they have been successfully used for the removal of polluting cation species such as ammonium and various heavy metals. Since 1990s, zeolites have undergone a notable evolution in two different directions towards higher pore size such as mesoporous silica, and also in the other direction towards higher specific surface area such as metal organic frameworks. But mesoporous materials are typically used after functionalization, and so there are a lot of possibility using a different functional group for the modified properties and catching different species with mesoporous. Metal organic framework are used a lot, but still very expensive. So for this reason, the research on zeolites has continued, and typically due to the negative charge on the framework, zeolites are cationic exchanger and usually have no affinity for anions and exhibit very low adsorption for organic in aqueous solution. But it's possible to change the surface properties by using, for example, organic surfactant. The absorption of anionic surfactant on the surface of the solids, for example, an quaternary amine as HDTMA, give rise to the formation of a bilayer and with a positive charge due to the functional group, such as in this case of amine, NR class, which acts as anionic exchange sites. So in this way, it's possible to modify zeolites properties, and the zeolites modified will be able to capture at the same time not only cations but also different kinds of anions and also organic compound. In recent years, surfactant modified natural zeolites have been extensively studied as promising material for the removal of emerging contaminants. Due to the industrial and technological development, we found in the environment high level of xenobiotic compounds and such as pharmaceuticals, steroid hormones, personal care products, pesticide, and others. And in the frame of an Hydrica project funded by European community through the Campania region, we have recently completed in our laboratories and setting up of 2 lab-scale prototypes. The first one is an reactive permeable barrier and typically used as a cost-effective technological for in situ groundwater remediation. The second one was an plant based on a column loaded with surfactant modified natural zeolite plus zero valent iron. It's possible with this plant to remove an ion from water, and we use this plant to remove arsenate and chromate. In addition, also remove organic compound through a decomposition by zero valent and iron. The properties of zeolites depend on the proportion of silicon and alumina content, namely the silicon to aluminum ratio. Low silica zeolites with a silicon to aluminum ratio less than 2 are typically hydrophilic and provide favorable characteristics of ion exchange with an excellent ion exchange capacity. On the contrary, high silica zeolites with a silicon to aluminum ratio up to several thousands provide characteristics for absorption of organic compound in aqueous solution. So high silica zeolites have been found to be effective absorbed for the removal of organic micropollutants, including emerging contaminants. What about the future? Is very the future. There is a problem with the slide.
Now it's fine, please.
Okay, now maybe can you hear me?
Yes, please, please. Go ahead.
Okay. Now, zeolites can be used in different forms, as a powder in batch reactor, granules, pellets, beads in a column reaction, but also as a membrane, but also as monoliths. Monoliths are typically self-standing materials, and we try to produce zeolites monolithic by geopolymer gel conversion. It's possible to produce a geopolymer gel precursor by mixing metakaolin, silicon powder, and sodium hydroxide solution. The geopolymer conversion also generates an hierarchical porosity characterized by micro, meso, and macro porosity, and is possible to generate the macro porosity using the silicon powder that reacting developing hydrogen, which promotes an in situ inorganic foaming. It seems that the porosity generated by foaming process promotes the formation of zeolites mainly on the surface of the pore inside the structure, whereas the stratas remain typically amorphous. The specimen obtained here, for example, after 3 days of curing, showed the presence of two distinct zeolites, typically zeolite 4A and zeolite 13X, used as a softener commonly in detergency. For this reason, we tested the zeolites monolith for softening of tap water, and crystalline zeolitic surface of the pore we found is able to exchange calcium and magnesium with fast kinetic and high affinity. Recently, we propose also and multifunctional porous materials for wastewater treatment, and material based on diatomite foam with an hierarchical porosity already produced and characterized to be applied in building as well as aerospace sector. We evaluate the effect on the water purification efficiency of the addition of different carbonaceous nanofillers such as graphite, graphene, and graphene oxide. Among the nanocomposites products, we found that graphene showed a significantly improved absorption capacity towards cadmium respect to the only diatomine form. For this behavior is typically connected to the synergistic effects of the finer morphology of the powders and also of the porous materials. The higher foam surface and the highly exfoliated fillers, which permits a better dispersion of the filler in the matrix. The latest tendency of the scientific community regards the development of different classes of green materials able to solve pollution problems caused by industrial and human activity. Diatomite and chitosan, for example, used as a composite, were recently used to produce a broad spectrum of hybrid adsorbents, either in powder or in monolithic form for environmental pollutant removal. Their absorbent capacity toward anionic dye indigo carmine, for example, was evaluated. Data showed that chitosan improves the uptake of dye in both diatomite and chitosan system, as exploiting a synergistic effect due to the large specific here available in presence of diatomite and to the contribution of amino groups of chitosan, which interact with the sulfonate group of dye. So, in conclusion, I try to go at last slide. Although zeolites have had an evolution since 90s, it still remains a class of nanoporous materials of great technological interest. Natural and surfactant modified zeolites, low and high silica zeolites, synthetic but also sometimes natural. Zeolites membranes, also another technology that was studied a lot in the last years, are just a few examples of zeolites with real potential application in the water treatment. But in the future, interesting innovation for the removal of polluting species from water, and in particular the emerging contaminants may arise from the research started in the recent years on monolithic and multifunctional porous materials. And so see you at next edition without the problem of audio and video for the New Time conference to talk about it. Thank you a lot and sorry for the problem.
Thank you, Domenico. Of course, we hope to meet in person in the next edition of New Times conference. To meet all of you in person.
I have to improve the
Discussing
connection.
face to face is much more easier.
We off without connection by internet in the presence. Yeah, of course. Thank you so much.
I'm sorry, a little bit late on the schedule. So I think we have time just for a very quick question. And of course, quick answer. I don't know from the audience, any curiosity or if not, I would like to know your opinion on, of course, it's my field, membranes. So on zeolite mixed matrix membranes, I don't know for water treatment, of course, are already used in gas separation. Can I have your opinion on this kind of devices for water treatment?
Thank you for your question. I don't have experience on membranes, but I know that Drioli in Calabria worked a lot several years ago. I remember Saro Aiello told me on the possibility to synthesize zeolites on the surface of ceramic support. I know that there are some application of zeolites in the separation water alcohol. Pervaporation is a typical sector where in the past was used, proposed zeolites in the membrane. I don't know the situation now regarding the reverse osmosis.
Yeah, I know that. I ask you if eventually this kind of devices can be used as a for softening purposes, no? Because
yes,
water,
of course.
in seawater desalination, for instance, we have problems with sparingly soluble salts, calcium carbonate, calcium sulfate, and so on, having very low solubility. So in this case, softening is a very important issue. Especially when you work at
it's possible to have a different stage before removing salt and after removing other pollutants. Is a typical possibility. Yes, the use of zeolites as a softening is very, very common. and so typically the outer detergents in powder typically contain 30% of zeolite powders. There are good possibility in the future. Yes, yes.
Okay, thank you, Domenico. Sorry for
no, sorry.
And thank you again. And we will move to the next speaker. Lia Vasquez from Italian Institute of Technology in Genova. The title of her presentation is PSA Composite Cryogels for Simultaneous Oil and Dye Removal. Please.
Good afternoon, everyone. I would like to start by thanking the conference organizers for providing the opportunity to present our work about the use of PSA composite cryogels for the simultaneous removal of oil and dye by gravity-driven filtration. These substances and other organic pollutants are currently a persistent and common problem that has induced severe water pollution. And this is primarily related to untreated discharges or unaffected wastewater treatments. Moreover, because oily wastewater can come from different sources, they might contain different pollutants that can make an integral treatment process challenging. Therefore, there is a need to implement innovative integrative removal process to achieve both oil removal and target other pollutants in oily wastewaters. The technologies for the removal of oily pollutants from water should preferably produce water of quality, treating simultaneously different pollutants, be easy to handle, facilitate the water management cycle, and be energy efficient and cost effective. Why hydrogel-based filters could be a solution? They might provide an alternative because they can be produced by several types of fabrication methods, obtaining different pore structures with desired wettability. Besides, because of their hydrophilic nature, they develop underwater oleophobicity, which favors the effective separation of oil-water mixtures, rejecting the oil droplets while allowing the permeation of the clean water. Also, the abundant active functional groups within the hydrogel network endows them with the ability to absorb organic molecules and anchor nanomaterials. However, there are a few problems with this approach. In general, one of the problems is that most of the hydrogel-based filters have a 2D configuration with short permeation paths that are prone to be fouled by oil during long-term operations. The other problem is that oil and dye are removed in separated steps, complicating the water treatment process. And last, after the absorption of the dyes, the cleaning of the filters involves corrosive solutions that might affect the filter's durability and pose environmental hazards. So, we think that a possible alternative to overcome this issue is the use of 3D hydrogels as filters, since they provide longer permeation paths. Also, these paths provide a high exposed surface area, promoting the absorption of the dye while allowing the water permeation. Additionally, due to the active functional groups, we can make grow in situ particles with photocatalytic properties to clean the filter. For this reason, the aim of the work being presented today is the fabrication of 3D hydrogel composites capable of removing oil and dye simultaneously from water by gravity-driven filtration and with photocatalytic self-cleaning properties. To achieve this, we have chosen PSA cryogel as a polymeric matrix because it has a superhydrophilic porous 3D network. Also, it has a high ion exchange capacity that makes it suitable for the synthesis in situ of particles. And we have chosen zinc oxide as a photocatalytic agent to provide the cryogel with self-cleaning properties. We first fabricate a polysodium acrylate cryogel by mixing all these elements for their polymerization reaction in a semi-frozen system, in which The ice crystals serve as pour templates. So after we fabricate the cryogel, we dip it in a zinc precursor solution and add sodium hydroxide for the in situ formation of the zinc oxide particles. As you can see in the same image, the unmodified cryogel presents a highly porous and well-interconnected network. After the functionalization, it is possible to observe bright spots on the surface of the cryogel, suggesting the formation of the zinc oxide particles, which appear not to affect the morphology of the cryogen. In the image taken at higher magnification, it is evident the formation of the rice-shaped particles of about 1 micrometer. The EDS elemental mapping of the functionalized cryogel shows the superficial deposition of zinc, while the XRD analysis confirmed the formation of crystalline zinc oxide particles. In this video, you can observe how the PSA cryogel absorbs water very fast, reaching the equilibrium in less than 15 seconds. It can swell 20 times its initial volume. However, after the incorporation of the particles, this swelling degree decreases by about 50%. The water permeability follows the same tendency, showing lower flow rates for the functionalized cryogel. Regarding the underwater oleophobicity, when the PSA cryogel is immersed in water, it shows underwater super oleophobicity. And also, we found that the addition of the zinc oxide particles enhances the underwater oleophobic properties of the unmodified cryogel and shows lower oil sliding angles. Then, we test the cryogels as filters for the separation of oil-water mixtures. The filtration flow rate of the PSA cryogel is about 20,000 liters per meter square per hour, while the modified cryogel is about 10,000 liters per meter square per hour. The oil separation efficiencies of both cryogels are higher than 99.97%. We have also evaluated the ability of the cryogels to remove the methylene blue from an aqueous solution during the filtration process, and we found that both cryogels develop high flow rates and separation efficiencies higher than 98.3%. The high separation efficiency is due to the electrostatic attraction between the negative charge carboxylic groups in the cryogel network with the cationic methylene blue. Next, we test the performance of the cryogel for the simultaneous removal of oil and dye. Here in the video, I showed you the filtration of the multi-component mixture using PSA cryogel as a filter. As you can see, the initial mixture has a light blue color. However, after we put it on top of the cryogel, clean water permeates within few seconds. In the meantime, while we wait for the filtration to end, let's have a look at the results. As shown in the graph, both foams can perform rapid filtrations while simultaneously remove oil, achieving a separation efficiency higher than 99.99%, and removing dye with a separation efficiency higher than 99.14%. To the best of our knowledge, this is the first report of a cryogel filter that can perform simultaneous removal of oil and dye from the water with such a high efficiency by gravity-driven filtration process. As for the self-cleaning ability of the composite cryogels, we found that the foam zinc oxide particles have optoelectronic properties and an absorption age in the UV range. We found that under UV light, the methylene blue degradation increases with time. Also, the zinc oxide improved the self-cleaning properties of the pristine cryogel substantially, reaching methylene blue degradation of 51% after 24 hours. I would like to conclude my presentation summarizing the key points. First, we have shown the successful fabrication of underwater oleophobic porous structures by the cryogelation process. Next, we have demonstrated the successful synthesis in situ of zinc oxide particles formed on the PSA polymer matrix. Then, we evaluated the performance of the cryogels by the separation of oil-water, dye-water, and oil-dye-water mixture by gravity-driven filtration, showing high separation efficiencies and high flow rates. Finally, the composite cryogels have cell-cleaning photocatalytic properties, making possible the degradation of the soft dyes and their potential reutilization. As such, we can say that the PSA composites represent a simple approach to perform an integrated water treatment process. Thank you for your kind attention.
Thank you, Lia. Very, very, very interesting presentation, and now it's much more clear also the potential of cryogels for this kind of water treatment. There is time just for one question. Unfortunately, we are a bit late. Is anyone from the audience?
Yes. I have a curiosity. Can I?
Yes, please.
Have you tried to test another kind of dye so to verify the electrostatic interaction between the dye and the material?
For now, we are trying only with we choose the methylene blue as an organic pollutant to do this test, but we are considering to test other type of pollutants and also other type of dyes, even though not cotton.
Thank you.
Well, let's take a few seconds more if there is any other question.
Yes, can I?
Yes, please.
In the hybrid material you performed, zinc oxide is embedded into the polymer matrix, is not covered, is embedded inside. It's right?
Actually, it's not embedded. I think that it's mostly on the surface because it's intermatrix synthesis. So, after we form the cryogel, we make grow the particles on the surface of the cryogel. It's not. It's, for example, if we mix the already formed particles with the cryogel during the polymerization, it's different. Maybe we can have these particles inside the matrix, but in this case, they are mostly on the surface.
Okay, thank you for your response.
Well, I don't want to lose the opportunity of a so reactive audience. So let's check if there is one more curiosity to satisfy, one more question. Of course, please, very quickly.
Yes, I have a question. If I well understood, you exploit the weight of the water to feed through the hydrogel your feeding solution. So my question is, when the height of the column decrease, you decrease the driving force. So how did you evaluate the flux?
How do we evaluate the flux? Okay, we measure the time and the volume because we filtrate, for example, 15 ml, and we check the time that this filtration takes, and also we consider the area of the filter because in this case, this filter only has 6 millimeters diameter. It's very small. So, and with this size, we can reach these high flow rates that higher than 20,000 liters per hour.
Okay, thank you.
Yes, okay. Lia, thank you so much. I think that we can close here your this discussion, nice discussion. And okay, thank you again. And let's move to the last speaker of this part of the session, Erika Yvette Cedillo Gonzalez. Sorry for the pronunciation, from University of Modena and Reggio Calabria. The title of this talk is Towards a Plastic-Free Ocean, Green CO2-based photocatalysts for mitigation of micro and nanoplastic marine pollution. Please, Erika.
Good evening. My name is Erika Cedillo, and I am from University of Modena and Reggio Emilia. As you may know, currently our oceans are full of plastic. Plastic pollution has several negative consequences for the environment. For example, big plastic items cause entanglement and asphyxia of the marine biota. The smaller plastic debris, known as microplastics, when having diameters between 1 micrometer and 5 millimeters, or nanoplastics when they are even smaller, are often consumed by the marine biota, entering into the food chain. As microplastics and nanoplastics absorb toxic pollutants in their surfaces, they also introduce those chemicals into the food chain. Microplastics, they had been already found in the human body. If you are still wondering how microplastics finish in the ocean, let me tell you that currently it is known that wastewater treatment plants are the main sources of microplastics to both aquatic and terrestrial environments. More disturbing is the fact that currently our oceans behave not only as a sink for microplastic pollution, but also as a source. The sea spray from the waves is reintroducing microplastic into our atmosphere, and so, thanks to this phenomenon, plastic now spirals around the globe. So, due to the previous information, the best strategy to fight microplastics pollution seems to be the reduction or even the elimination of the microplastic inputs into the ocean. In this research, we propose the use of photocatalysis to achieve this goal. As photocatalysis generates highly oxidant species, we tested if a photocatalytic system is able to degrade such a complex pollutants like microplastics and nanoplastics. As you may know, photocatalysis respects 2 of the 12 principles of green chemistry. To make the process as sustainable as possible, we also tested the use of renewable raw materials to obtain our semiconductors. Particularly, we used the extrapolate fluid of seawater methylacetyl mussels. The original idea was first supported by the research group of Zeng and collaborators. In that work, the authors used freshwater Cristaria applicata mussels to develop a titanium dioxide material modified with nitrogen, with high surface area and active invisible light. That material was used to degrade water-soluble pollutants. So, why to use extrapolate fluids of mussels? The answer is to obtain porous photocatalysts. Usually, this kind of semiconductors are obtained by the use of copolymers that act as pore-forming agents, but copolymers are produced from raw materials derived from petroleum. Instead, the extrapolate fluid of mussels contain proteins, which can be used as renewable raw material, also as pore-forming agents and doping source. We prepare our materials following the procedure reported by Zeng et al. With some modifications. The first step was the collection of the extrapolate fluid from the methylacetyl seawater mussels. Then that extrapolate fluid was cleaned by centrifugation, and the cleaned liquid was mixed with the oxide precursors. After stirring and aging, the mixture was hydrothermally treated and yellow or brown powders were obtained. We obtained 2 materials. The first one was titanium dioxide modified with carbon and nitrogen. If we compare the properties of this material with those of the material obtained by Zeng et al., the main differences are that our material was modified not only by nitrogen but also by carbon, and we obtained a material with higher surface area. Our porosity type was mesoporous, and maybe the most important finding was that while Zeng et al. Needed to need to purify the extrapallial fluid of Cristaria applicata muscles, we only need to clean the extrapallial fluid of Mytilus muscles. So we were able, by using this muscle species, we were able to eliminate one step of the synthesis procedure. In the case of the mixed oxide, we found similar properties. Our material was modified not only by nitrogen but also by carbon. And also in this case, the extrapallial fluid was used as extracted without concentration procedures. Remarkable, the surface area of this material is 2.5 times that of the Zeng's material. This is the experimental setup we use for photocatalysis. We use aqueous medium because of its relevance for the wastewater treatment plants, and our main degradation monitoring tool was gravimetry. In this slide, the photocatalysis of polystyrene micro and nanoplastics is presented. The semiconductor that we used for this test was the titanium dioxide modified with carbon and nitrogen, but this material was deposited over glass substrate to obtain films. If you see the deep plot, you can see that even if we obtain low degradation values, we found that the process is possible and that decreasing the size of the pollutant from 1 micrometer to 300 nanometers, the initial degradation rate increases. As using the photocatalyst in the form of films do not fully exploit the properties of the materials, these were used in form of powders. Additionally, polyethylene microplastics were used because they are one of the most common microplastics found in the ocean. As you can see in this table, we reach degradation of microplastics of 70%. But that high degradation was achieved at very harsh conditions. The site of the pH 3 is explained because the photodegradation of plastics is favored at low pH. But how can we explain the effect of this very low temperature? Well, as you can imagine, when microplastics are exposed at low temperature, they freeze. When they freeze, they become brittle and break down into smaller pieces. By this decrease in particle size, there is an increase in surface area, and then there is a better contact between the very tiny plastic particles and the semiconductor powders. So, also in this case, we confirm that decreasing the size of microplastics increase the degradation of this kind of pollutants. The degradation of polyethylene microplastics was confirmed not only by gravimetry but also by optical and electronic microscopy and thermal analysis. For this specific system, we use scavengers to determine... Slide 19, definitiva. For this specific system, we use scavengers to determine which species participates in degradation. We found that the hydroxyl radicals derived from the photogenerated electron, as well as the hole and the superoxide radical ion promote degradation, as shown in this scheme. This was explained by the fact that due to their huge size, microplastics do not absorb on the semiconductor, but rather the semiconductor absorbs on the microplastic surface. So this phenomenon limits the role of the hydroxyl radicals that are absorbed in the surface of the semiconductor. And the process seems to be promoted by the free hydroxyl radicals derived from the photogenerated electron. The mixed oxide was used to degrade PET microplastics. We found that this material can work at less harsh conditions, but we also think that it depends of the particle size of the microplastics, as in this study, PET microplastics of diameters lower than 500 micrometers were used. As you saw, we were not able to reach complete degradation. However, we know that plastic photodegradation can lead to all these compounds that can be used as carbon source for several microorganisms of industrial interest. The conclusions of this work are the following. The extrapalate fluid from methylocellulose seawater mussels can be used as renewable raw material to obtain titanium dioxide-based photocatalyst. The proteins from the extrapalate fluid act as doping agents and pore-forming agents. Microplastics of polystyrene, polyethylene, and PET can be removed using the methylocellulose-derived green photocatalyst in visible light. Terresterna oyster is able to modify titanium dioxide by incorporating nitrogen. The materials are able to degrade polyethylene microplastics. However, there is still a need for research in this field, as many parameters of the reaction should be investigated to improve the photocatalytic removal efficiency. Indeed, this technology can be easily adapted as a tertiary treatment in wastewater treatment plants, eliminating or even reducing microplastic pollution of the effluents before they discharge into the aquatic environment. The application of photocatalysis technology can thus contribute to the protection of the ocean from microplastic pollution. Thanks to all of you.
Thank you so much, Erika, for your nice contribution in a so hot topic. We are consuming the time for the coffee break, so probably time for very quick few seconds of question and very rapid answer, if any.
Yes, I have one question. I missed the one point. Why do you use silicon dioxide?
Because I want to increase the surface area of the nanomaterial. So I found that when I mixed titanium dioxide with silicon oxide, the surface area was increased.
Okay.
Okay, thank you so much. I'm very, I'm sorry for this, but we have the coffee break, so we will start again in less than 10 minutes. Okay. Thank you to all the speakers and to the audience for the kind attention and the nice discussion. Thank you.
Yes, now the
I would like to
announcement of the awards.
Yeah, the organizing committee for inviting me and giving the honor to co-chair this session. I'd like to thank all the speakers for the very high quality of their contribution and all colleagues for the fruitful and interesting discussion. So I give the microphone to Antonio for, I assume,
First, I wanted to thank all the speakers of this session. We are very impressed by the scientific quality of all contribution, both oral and e-posters. Thanks to the support by Assing, which is particularly mindful of the relevance of environmental sustainability and ecological transition, we have 2 awards for young scientists participating to this session for the best oral contribution and the best e-poster, where e-poster is the usual poster with a supporting video of just 5 minutes. To give the Commission the possibility to complete the evaluation process to select the best oral contribution, we will start with the awarding to the best e-poster. Then we will broadcast the supporting video. We will come back in 5 minutes with a very short question time in order to give the audience the opportunity to interact with the winner. And soon after, we will announce the winner of the best communication. So let's start with the announcement of the best e-poster on environmental sustainability. The Commission of the New Times, New Trends in Material Science and Engineering Conference, considering the scientific quality of all e-poster, is glad to announce that the winner of the best e-poster in environmental sustainability is Marco Aquino from University of Calabria, with his contribution on low-cost cation exchange membranes for reverse electrodialysis. So now we will have the broadcasting of 5 minutes of supporting video, and then we will come back with Marco. So and soon after, we will announce the best communication. So let's start with the video.
Good afternoon, everyone. My name is Marco Aquino. I am a PhD student in Environmental Science and Engineering of Construction and Energy at the University of Calabria. First, I want to thank everyone for the opportunity to participate in this conference, and today I will talk about the work low-cost cation exchange membrane for reverse electrodialysis, preparation, characterization, and optimization under the supervision of the Professor Ephraim Kurcho. The aim of this study was to optimize the electrochemical properties of the CEMs in order to utilize it in reverse electrodialysis operation. Reverse electrodialysis is an emerging technology able to convert the Gibbs-free energy of mixed solution into electric power, thus exploiting the salinity gradient established between 2 solutions having different concentration. Reverse electrodialysis based on the use on ion exchange membranes, and one of the main limitations to the commercialization of the reverse electrodialysis is the high membrane cost. Sulfonation of low-cost commercial polysulfone was carried out in order to graft negatively charged groups onto the polymer structure. Sulfonation was confirmed by FTIR, which show characteristic peaks of sulfonation. The graph transmittance wave number show the FTIR patterns of polysulfone and sulfonation polysulfone in the range for sulfonation polysulfone footprint. Characteristic symmetric and asymmetric stretching sulfonate peaks were obtained at 10, 30 and 10, 19 per centimeter in the first sulfonation, and in the second sulfonation at 1030 and 1100 per centimeters. In the subsequent step, cation exchange membrane were prepared by using the sulfonated polysulfone. Two methods of membrane preparation was used: immersion precipitation in sodium chloride solution to obtain a porous membranes, and solvent evaporation to obtain a dense membranes. Sulfonation polysulfone with a sulfonation degree of 0.48 was chosen to prepare the membranes, as polymer with high sulfonation degree resulted in poor mechanical properties. The preliminary results obtained from the characterization test of the membranes made with sulfonation polysulfone are discordant. The membrane made with the phase inversion method show low resistance but also zero permeselectivity. The membranes made with the evaporation method show a good permeselectivity but a very high resistance. Membrane S2 was the starting point for the optimization for these negatively charged CMs. In fact, I prepare a more open membranes in order to decrease the resistance, but without losing the permeselectivity value reached. The best properties, permeselectivity of 0.89 and electrical resistance of 1.39 ohm centimeter square, were obtained for the membrane prepared with the solvent DMF, co-solvent IPA, polymer sulfonation polysulfone in ratio 67.5, 7.5, and 25. The application of this strategy allowed to optimize the electrochemical properties of the S2 membranes. Cheap and easy scalable sulfonation and membrane preparation procedures at lab scale, price of chlorosulfonic acid and polysulfone are $0.56 per kilogram and in between 2 and $7.5 per kilogram open new path for the practical implementation of reverse electrodialysis.
Thank you, Marco.
Thank you so much for this honor. First, I would like to thank the New Times Committee for organizing this very interesting conference at this particular time. Activate the video.
I have a short question for you. Your poster deals with reverse electrodialysis, which is particularly relevant for ecological transition due to the possibility to get blue energy. So, do you have thought how to combine reverse electrodialysis with the platform of solar technology? So, with photothermal membrane distillation as an example.
Yes, yes. I am moved and honored by this award received. In this preliminary study, I concentrated on the preparation and optimization of the cheap cation exchange membrane for reverse electrodialysis. For future works, the idea is to optimize the membrane technology at the water, energy, raw materials nexus, combining two technology such as reverse electrodialysis and membrane distillation with the reverse osmosing allow at increasing the water recovery factor, reducing the environment impact for discharge brain, and recovering the electrochemical potential for the brain. This is possible with the advent of photothermal assisted membrane crystallization. In fact, preparing and using thermoplasmonic membrane on the basis of the circular economy, they recommended that is produced by membrane distillation and membrane crystallization process can be used in the reverse electrodialysis technology to generate the energy.
Yes, thank you. This should also enable the possibility to get an autonomous solar desalinator, exploiting both sunlight and the energy produced by reverse electrodialysis. So, thank you again, and let's move to the announcement of the Assing Prize for the best contribution in environmental sustainability. So the quality of this session was really high, but the Commission has decided to assign the prize for the best contribution in environmental sustainability to Lia Vasquez from Smart Materials, Fondazione Istituto Italiano di Tecnologia in Genova, for her contribution, polysodium acrylate composite cryogels for simultaneous oil and dye removal. So congratulations to Lia. Thank you very much for having joined this conference with this interesting contribution. So, the floor is yours.
Thank you very much.
So, we can conclude this session, and thank you again also for having remained connected up to this time. And let's see tomorrow morning for surface science. Congratulations again to Marco and to Lia.
Thank you very much.